Hi everyone, my name is Sania Koller, and today I want to talk to you guys about some interesting problem in bioinformatics, which is how can we use mutual information to infer transcriptional gene regulatory networks. So how can we use mutual information to figure out and use network biology to figure out which transcription factors are regulating which genes and what is the connection? And that is usually how we can determine the impact of various genes and the relationships between them. That is really important when trying to better understand our body and causes for diseases and pathways and just contribute to the branch of science in general. And please don't worry if you don't understand some of these terms, I will explain them. And of course, you can always write questions and ask me any questions you have along the way. So this application is in bioinformatics and in genetics as well, but it's also a computer science concept of mutual information. So typically we have these genes. The genes are regions along the DNA, which encode usually a polypeptide, and that's becomes a functional protein or part of a protein. And so here's a gene along a piece of DNA. And what transcription factors are that they're proteins. They help regulate the expression of genes, either the activation or the repression of genes by binding. So along the DNA, they bind to these sequence-specific transcription factor binding sites. So you can think of them like puzzle pieces. And these are usually in regions like promoters or enhancers, and they bind to these regions like puzzle pieces. T and A bind, A and T, C and G, G and C, T and A and T, C and G. And it's just like puzzle pieces. It's really cute, right? So it's sequence-specific motifs. And when they bind there, they help regulate the expression of that gene. And that also involves bringing the RNA polymerase II and other factors, but it's really important that these transcription factors are able to bind to these transcription factor binding sites, and then they are regulating the expression of their target genes. So in general, the central dogma of molecular biology is as follows, and credit to Professor Daifeng Wang from UW-Madison for this slide as well. So we can have identical DNA but different gene expression. In this nice movie on twins, it's about epigenetics, nature versus nurture. But in general, the central dogma of molecular biology is that we have a piece of DNA and we are transcribing that, we find a gene and we're transcribing that gene, into messenger RNA, and that messenger RNA is then leaves the nucleus and gets after some pre-processing steps, and it then gets translated into protein. But what is a key part in this process are transcription factors. Transcription factors can lead to whether or not a gene gets transcribed in the first place from a gene to a messenger RNA. And what we can try to do is we want to figure out what is the expression level of a gene and how is that connected with the disease that we observe. So we can look at nerve cells, bone cells, look at the different gene functions of these genes. And gene regulation is mechanisms that control the expression of various genes. So why do we observe more genes in certain cells than in others, for instance? Or in diseases, why do some genes go haywire? How do some cells rapidly divide a lot, in cancers? Like, what is happening? So at this level of transcription from, gene to RNA, from DNA to RNA of the gene. So we have DNA for the gene to messenger RNA. There are many factors here that are involved in this process of transcription. And as the name suggests, that's also the role of these transcription factors. Transcription factors can play a key role in the regulation of this gene, like the transcription of this gene from DNA to mRNA, and then basically the next part here is translation to a functional protein. in this step here, and this is the translation by the ribosome. And this, that's what this translation part is over here. But basically, like what we're focusing on here is we're trying to infer a transcriptional regulatory network that's going to try to tell us which are some of the transcription factors that are kind of responsible for the regulation of this gene. So that will give us a better understanding as to what goes on in terms of the gene expression.
So hi guys, so basically what this is known as is the central dogma of molecular biology again. So basically how do you go from DNA to RNA, by transcription, to messenger RNA, which you see the poly A tail, and then this is all within the nucleus of the cell. Then you do some pre-processing as well, where introns get removed, you join the exons together, you export it. And then you translate it with the ribosome. So you turn these into amino acids, and usually they lead to a protein or polypeptide chain. So this is the process that we're looking at here. And what we are really concerned with right now here is, this part here, which usually involves, transcription factors. Or TS. They're kind of involved in helping with the transcription in general. And this ultimately, we're trying to figure out, okay, well, why is there a lot more of a certain gene in the cell than another type? So what we're really interested in is trying to analyze what's going on over here. So again, the transcription involves multiple different factors. And then that leads to the DNA, becoming, we're looking at a specific segment here, like a gene. Like we could transcribe this, and then into like this region of interest will be transcribed into messenger RNA, which will be translated into amino acid and then protein. So there are multiple reasons why we can observe the gene expression levels at the way they are. And here, what we've been focusing on a lot is like, focusing on factors that are involved in this key step over here. And these are transcription factors, that we're looking at. So pretty much, you have the transcription start site, and then this is for like your gene of interest. We're trying to go from, from our gene, and that gene will ultimately get expressed in the form of a protein. It'll be part of a protein, or it'll make one protein for sure. But what we're interested in is seeing, okay, that this is the gene here. And then we have a core promoter, we have a Tata box and proximal promoter, distal regulatory regions like enhancers, silencers, and then we have gene regulatory proteins, co-activators, RNA polymerase, and general transcription factors. So we're interested in like these transcription factors that are binding here, recruiting RNA polymerase, and then that leads to the transcription of a gene. This is the transcription start site. This part here is what we're trying to tie in, like what's happening here. This overall picture of what are all the factors that are involved in helping with the transcription of a given gene. That's sort of what we are interested in trying to figure this out. So overall, what we've been talking about is like gene regulatory network inference, and like we looked at sequence specific motifs, where you can look at the height of the DNA bases in given positions, figure out where transcription factors would bind along a set of DNA, and try to make those inferences. So we're going to talk some more about how we can look at the mutual information between them to try to figure out like gene regulatory networks.
And again, these transcription factors, and credit again to Professor Wang, these transcription factors bind to the sequence-specific motifs, and we can use, frequency logos or information content logo as well, which is also based on entropy of the random variable that represents a distribution of states in each position. And here, the height here represents how certain we are of a given position. like here, for instance, in this last part in this motif, we really see that there is a pun intended, actually, that's a pun. We really see that there is like a 70% chance of C. So there's really a lot of information there. I mean, so that has a higher height, because it's very prevalent, but also we know that with 70% certainty that we're going to observe a C. So that's why this height is much greater, because we have more information about this last position. We have a higher probability that it is a certain base, so there's less uncertainty. But when it's more uncertain, like here in the second position, where it's a 20% chance or 30% chance for given DNA bases, it's like more uncertain. It's almost like, it could be anything. We don't really know. So that's where they're roughly equal height here. And over here, there's virtually like no information that we really have from this. So we don't really know what it could be. We don't really have that benefit. It's like if somebody already tells you something, like if if you find out, okay, this is what, if you open up your Christmas presents or birthday presents before Diwali presents or whatever you celebrate, and it's like you peek and look before, it's like you're not as surprised, because you don't really have, there's not much to surprise you. So here it's like it can be, this is what I kind of expected, like this is 20%, 30%. It's like it's almost like equal chances. So it's like sort of what I expect. So I didn't really gain much, from this, the information content. So when you actually, if you peek beforehand and you open up your presents on the day of, you're not really phased. You already kind of knew what it was, So I think that ruins the fun. So try not to do that, like don't peek, just wait for the day of. And kind of what I want to do is also like go over a quick review of some network biology concepts to try to explain what are transcriptional regulatory networks for you guys. And now a key part of transcriptional regulatory networks is that it tries to explain how these transcription factors are involved in the regulation of certain target genes. So here we can have 3 different transcription factors, MAFF, IRF1, and IRF9. So this is one of the diagrams I had built once using data when I was public data related to COVID, for instance. And here is interleukin 6, which is one of the immune system related interleukin molecules. So what you see here in this transcriptional regulatory network is that we're trying to explain how these transcription factors are involved in regulating the expression of key genes. So for instance, the way that it typically goes is you have a transcription factor, the TF, a transcription factor, and this transcription factor is involved in the regulation of a target gene. So here, this would be like a target gene. So it's involved in the expression of its target gene over here. So that's sort of what's really important here. So here, the squares are the transcription factors. So we have these 3 transcription factors, IRF1, MAFF, and IRF9. So these are special types of genes, like, proteins that are involved in regulating the expression of these target genes here. You can see all 3 of them have at play that they're regulating IL-6. And some transcription factors also regulate one another, so they, co-regulate each other. And so whenever you see an edge, each of these edges here, it's representing that the transcription factor is involved in regulating this target gene. So here what this is showing is that if it's negative, if it's repression, where the transcription factor is reducing the expression of a target gene, then we would expect to see these blue edges. It's blue edges. Again, they are associated with repression of gene expression in this diagram. So this blue part here is repression. So MAF is reducing the expression of COX-15. That's based on this. This is a negative number, negative, and the positive number here is meaning activation. So all of these refer to activation, where the transcription factor is helping activate the expression, so it's positive. And this is negative here because Repression means that it's leading to the loss of the expression of this gene. So basically, again, just to clarify that this part here is repression. This is negative regulation. It's associated with negative expression or repression of that gene by the transcription factor, and positive regulation is associated with activation of a gene by a transcription factor, and this is a directed network. So you can see that, there are different tools as well, like, GeneNet is a web server for gene network construction and visualization as well. And you guys can check this out as well. It's really cool. And then there are other types of networks where there's undirected, so there's no directionality in the arrows, and these are gene co-expression networks. So you can see that like sometimes it's also like a hairball, and they all are co-expressed. They share similar expression dynamic patterns. And usually these are correlation networks, and there are multiple different software and algorithms like weighted gene co-expression network analysis, WGCNA, which I love, as well is one of the popular approaches to build these networks. Other ones are Gaussian graphical models, and they can try to explain like tissue-specific control and other links. And usually we come up with modules, groupings of genes that are very similar, and they have similar functions, similar expression dynamic patterns. So they're just a group of genes that are highly correlated with each other. And then we can perform enrichment analysis on what does this module of genes do. Like for instance, if these are all highly correlated with each other, like they might have some function. Like let's check out like a bunch of sources to see what those functions could be. So again, these are undirected. But for transcriptional regulatory networks, we know that the transcription factors are helping control the expression of the other genes. But here we may not know necessarily if any are like controlling the expression or helping regulate it. It's just going both ways, from a gene to another gene. So it's just like the way that it looks is just like, you have a gene A and then you have a gene B. Oh wait, I should probably show it a little bit. We have, like, let me just write this out. And then what we also have is so each of these circles is representing a gene, and here, because it's both arrows. Let me draw that properly. And let me just make it a little bit proper. Gene A and gene B. So it's just the same thing as, arrows going both ways. So it's bidirectional. There's really no edge direction. So you can also say that in essence, what this is just saying is that the directionality is just it's bidirectional. So that's how you can interpret these co-expression networks. And again, like these can be like, hairballs, and it's difficult to learn such networks. So it's like a clump of hair, I can imagine just, like a clump of hair on the hairbrush, for instance. Like that's how these networks look, with so many edges, so many connections. Sometimes it's very difficult to infer these networks, and we need to use like regularization parameters.
So basically, what we've been talking about are these different networks in network biology, like co-expression networks for different genes, how they interact with each other, what are the relationships between them. And we can sort of extrapolate that to protein-protein interaction networks as well. The relationship between different proteins, how they're interacting with each other. So kind of like our main focus here is going to be on gene regulatory networks, also known as transcriptional regulatory networks. So again, these are really direct. You see the directionality, the arrows pointing. This means that this gene here is involved in regulating this transcription factor, and then this transcription factor is involved in regulating this gene. There might be some sort of co-expression there, but in general, it really should be going from transcription factor to gene. So what you'll observe here is that we have TF, we'll have the and then we'll have the regulated gene. So this is the relationship that you're going to find, transcription factor and then the gene that it regulates. You know, by binding to that enhancer region, promoter region for that gene, it's involved in the regulation of this gene. So it's really, you know, this can be an activation in a way. So it's like, you know, it's a positive interaction. And then this, it typically looks like a stick in a way. It looks a little bit like this. This means like it's more of a repression, like it's a negative interaction. So it's a negative interaction, but it's like it's kind of inhibiting the expression, and this is more of a positive interaction here. It's a happy smiley. So these are gene regulatory networks, also known as transcriptional regulatory networks, and these are protein-protein interaction networks. Or we can also talk about, in a way, you know, gene co-expression networks. Like genes that are co-expressed together, like we were looking at before. And what we'll see here is that, like, for this gene co-expression network, that's a protein-protein interaction network, these are proteins or peptides, and they just co-express. There's no directionality between them. We're not sure like what is regulating what. And this is really great for Bayesian inference as well here. And just sort of wanting to talk a little bit more about, you know, the gene regulatory network or transcriptional regulatory network again. So you can imagine you have gene 1, you know, trying to make messenger RNA 1 for this gene, and then you know, it becomes protein 1. That protein 1 you know, can do many different things. You know, the genes, it's like a chicken and egg problem, like who came first, the chicken or the egg, right, in a way. Like, you know, even the universe questions in a way. So this gene, it's come and it's involved, it's produced mRNA, and this mRNA becomes protein. Remember, we can go from gene to protein. And then this protein can be doing multiple things in the network. So this protein can be binding as a transcription factor potentially to be involved in the regulation of this other gene in this network. So, you know, this gene here could very well be a transcription factor. So in this case, it's a transcription factor. So it's got many things. So it's a transcription factor for gene 2. For gene 2, that's what's happening over here. But it can also be the transcription factor in a way for gene. It can also be involved for gene 1 as well. Gene 3, so for gene 3. Let me make this color a little better. It can also be the transcription factor for gene 3. That's what's happening over here. Because if we track what's happening for gene 1, what we can observe that's happening is gene 1 makes this protein. This protein helps bind to gene 2. And gene 2 then produces, gets activated. So it's a transcription factor. It's binding to like a promoter or enhancer in front of gene 2. Then gene 2 is producing a mRNA, and that mRNA leaves the nucleus for this gene 2. And gets the ribosome, then works on it and turns it into a protein 2. And then this is the protein that corresponds to gene 2. So this is all the central molecular dogma of molecular biology, makes this gene 2 become protein 2. Then what happens is that these protein 1 and protein 2 then are needed together. They're needed together. You need these together to then activate gene 3. So gene 3 needs gene 2 and gene 1 to bind here, and then it can be transcribed into messenger RNA and then protein 3. So what you're seeing is that gene 1 is needed by gene 2. And gene 1 is also needed by gene 3, and gene 3 also needs gene 1 and gene 2. So gene 1 needs gene 1 is needed by gene 2. So gene 2 needs gene 1 to bind here for it to get regulated. So that's what this relationship is showing. This is this relationship here. That's what's being shown over here by that relationship here. Then this protein 1 being a part of gene 3 regulation is what's shown by this orange highlight here. And maybe I should also show the role of, let me use another color just to show this, how this protein 2 is then being used in gene 3. That's what this part is measuring here. You can see like yellow, yellowish, bluish is like green. So this gene 1 to gene 3 again is how gene 1 is, the protein, the transcription factor, is being used to transcribe gene 3 to protein 3. And this part here shows how gene 2 is used for helping make protein 2. That then comes as well, and both of these are needed to express gene 3 into protein 3. So that's what this gene regulatory network is sort of trying to tell us, that gene 3 is determined by gene 2 and gene 1. So here we don't know its parents for gene 1, but gene 2 needs gene 1 and gene 3 needs both of these. The arrows are pointing from parent to child in this. So the way it always goes is it typically just goes from parent. It can be multiple parent, but for each parent, you go to the child. And this is usually the transcription factor regulated gene, or also known as target gene.
So going back to transcriptional regulatory networks, we can try to infer these. So a credit to Dr. Shishmita Roy from UW-Madison. So these slides are taken from her notes as well. So pretty much we have next generation sequencing networks, techniques for regulatory networks, DNA-seq, ChIP-seq, chromatin immunoprecipitation, RNA sequencing. We can use all these different data, to find these signals where transcription factors are binding and recognizing the motif along on the DNA and where they're binding. So we can try to figure out, okay, well, what transcription factors could be involved in regulating which genes. So that's what this is taking care of. And again, what a regulatory network typically captures is the relationship between, like, these are transcription factors and they're regulating this heat shock protein 12. So, like HOT1 is regulating, so the arrow is going from here to here, and SKO1 is also regulating HSP12. So if these transcription factors are binding, if this transcription factor and this transcription factor, if together they're binding to these regulatory sequences like promoters or enhancers, so this is typically non-coding DNA region. So if they bind there, that leads to the activation and expression of HSP12. So then it will be turned on because these are activating. So because these are activating, so if it goes in this direction here, where they're activating, so that's the directionality where we need these two transcription factors. So these are like the TFs, and then this is the target gene. The target gene is HSP12. So this is the target gene. So that's what this is showing as well. And if they're not there, these transcription factors here, if HOT1 and SKO1, if they're not there, then the transcription of HSP12 will not happen. So that's what these networks are trying to basically explain the relationship between the transcription factors and the target gene. So you can imagine this is a stretch of DNA where to have like this transcription here of like 3 different mRNAs, messenger RNAs, of HSP12, you need these to bind to those regulatory regions. So again, a transcriptional regulatory network, I've shown you an example. It has these transcription factors like A and B. They're binding to a regulatory element, like a promoter or an enhancer of this DNA. and that helps lead to the transcription of gene C. So this arrow pointing from A to C and B to C. So you need both of them here. So these are the transcription factors. So this is a TF. Oh gosh. And this part is your target gene here. So the way that you can see, again, it's regulated. There's a directionality. So it's a directed, signed, weighted graph, and nodes are transcription factors. And then they're the target genes. So this transcription factor may be regulated by other genes. These could even co-regulate each other, for instance. So A regulates C's expression level, and B also regulates C's expression level. And maybe these transcription factors are regulated by others, and it's like a chain reaction as well.
And just to reiterate it again, this is just another example of a transcriptional regulatory network. And here again is another network where you can see the genes that are involved in regulating other genes. And I just hope you can understand sort of what's going on behind the scenes. You know, like here GLI3 is involved in regulating PBR, and PBR is also regulated by GLI. And then GLI3 is involved in regulating FOXA2. And GLI2, like it just kind of shows all the relationships that are happening. And these here, for instance, are like transcription factors, and these are the proteins. As I was showing you before, that this little bar, it looks like it's a negative effect, which is more like repression. It's like repression or inhibition. And this positive effect here is just like more like activation. So these are all like important things to note. This is transcriptional regulatory network here.
So basically, one of the ways that we can also infer transcriptional regulatory networks is by something that's known as mutual information. So that's called MI. So what you often learn about is that there's mutual information that we can use to figure out what are these transcriptional regulatory networks. And on Bioconductor, there's a package that's really popular that I really I've used as well, and I think it's great. It's called RTN. So what this does is it builds a transcriptional regulatory network using data such as gene expression data, like RNA expression, where your data basically looks of the form where you have a bunch of genes. It can be all sorts, but basically the way that the data looks is you have like it's high dimensional, so you have genes. You have genes here, and going across, you could have like samples, like individuals. Like you have samples, and then you have the expression level. So each of these is like an expression level, like that's what each data cell represents an expression level. And I'll show you an example as well of what that looks like. But pretty much what that takes is it takes that data from high throughput gene expression data. So that's what this is saying here. It'll take that gene expression data, and basically it's genes with something. Usually they can be patient samples as well, like people with the disease, like each individual. Maybe they're a time point, for instance. And what they typically do is they take this in, so the gene across the samples, and they're going to try to infer a transcriptional regulatory network like this, for instance, where, again, this is similar to what I was showing before. So the image I'd actually made for my COVID public data side project was actually like using this RTN package here. And you have transcription factor E2, F2, FOXM1, PTTG1. Another thing to note is that Bioconductor are just packages that use RStudio as well, and they're for biology, and they are amazing. So this is R below as well. And this is a vignette, so it's a tutorial. So what it's trying to do is it's trying to test all the associations between a given transcription factor and all potential targets using either RNA sequencing data or microarray transcriptome data. So how exactly does it do it? Well, it looks at the mutual information. So that's what MI is. So it's computing mutual information between a regulator and all potential targets. So usually what you give in is you give in a list of candidate transcription factors. So genes that you think are likely to be transcription factors. So people are still like researching what they are, and there's like Jasper's list and there's Lambert's list of transcription factors. So then you have a named gene expression matrix, a data frame with gene annotations, and another sample annotations. But the most important thing I wanted you guys to see is that it's looking at the mutual information between a regulator. This is also another name for a transcription factor, and all potential targets, like all target genes. But it's using mutual information to infer this regulatory network. So that's why this is a very important algorithm. People are using this to figure out, okay, which genes are regulating other genes in disease, for instance. And a lot of biological applications, when you look at different networks, it's often like one gene is regulating another gene, and there's like this whole cascade of signaling pathways that's involved. So what this point here is to show you is that mutual information is really widely used in a lot of papers. Like this is a paper like benchmarking algorithms for gene regulatory network inference from single cell transcriptomic data. That's usually on the gene expression values within a single cell. So that's even like going inside, like for the brain, it could be like oligodendrocytes, microglia, astrocytes, like a lot of glial cells apart from neurons. Or like in the immune system, we have like all sorts of T cells. We have CD8 cells, which are cytotoxic T cells, or we have CD4 cells, which are helper T cells. We have like B cells, we have all sorts of, macrophages. We have so many different types, neutrophils. So that's like sort of like a more, like granular level. But the gene regulatory network, again, that's sort of like a transcription regulatory network that we talked about. They're interchangeable, like basically the regulation of genes. And you can see mutual information is one of them. Then there's like random forest correlation. So it's a very popular technique. And again, when we're looking at mutual information, we're trying to measure the amount of information that one random variable contains about another random variable. So, we're given two random variables like x and y. They have a joint probability mass function, so that's sort of seeing like how do their probabilities change together, and how like x and y together, and x individually and y individually. These are the marginal ones, and these are the joint ones. Joint is like their joint behavior, their distribution together. So what the mutual information I, x, y. So when we talk about mutual information, we usually refer to it as I of x, y. So that's like the mutual information between x and y. So these are two random variables. So the formula that we use is we typically look at all the values of x, all the values of y, and we look at the joint probability, p of x comma y. I mean, this means and, the comma means and then multiplied here within the same summation by this is like the log, this is a log probability of them. And under this log probability, we're going to be looking at the same joint probability p x comma y all the way over p of x. This is the marginal for x times p of y. That's a marginal for y. And then that's what this value would give us. In a way, that's what this is saying, that we just take like the values of x and the values of y, we multiply them by the means probability of x and y, then we multiply by log probability of x and y divided by probability of x and probability of y. And that's how we can sort of infer these transcriptional regulatory networks over here using this type of information. And we can try to reconstruct these transcriptional regulatory networks using many different parameters, including mutual information is just one of them. So yeah, we're going to talk about examples about all of that, so please stay tight. So again, like this is like, like a random variable x, random variable y, and what, credit to Professor Dife and Wang for this slide, but mutual information is just telling how much knowing the value of one variable tells about the value of another. You can think of, mutual information is like if you have like twins, knowing one of them is going to tell you the value of the other, for the most part. You can kind of predict, what they're going to look like. If a twin looks like this, the identical twin would probably pretty much look like that in a way, right? Or you can kind of get a lot of information, knowing some value will tell you something else, like these days we get a lot of mutual information from our gadgets, like our cell phones, because we always carry them, so we get a lot of information, like knowing where one variable like our phone is will probably tell us where we are, because we end up carrying our cell phones a lot. So again, this is the same thing as before, where instead you now have M and you have C. You have these two random variables, and you look at what this is mentioning again, is what this measures is this joint probability. This is the probability of M and C. And like you can also use Bayes' rule, where if you want to find what those are, then that probability would be probability of M given C times the probability of C. For instance, that's what this probability, so these things are analogous, this thing is and this thing are the same. This is M given that C happened, and this is the probability that C happened. And then you can think of it like this in a way, like when you have this and you're trying to figure out, okay, well, what's the probability of X happening given Y happening? Like if you know that Y has occurred, this whole thing has taken place, what's the probability that X will occur given that? So you're looking for this part here, this part, you're looking for this part out of this whole entire thing here, this whole thing. So that's what mutual information is, just trying to tell you how much does knowing one variable, its values, how much does it tell you about the value of another in a way. And this is sort of like a really nice show, Sister Sister. It takes place in the 90s, late 90s, so I'm currently watching it. It's very nice. I really like it. So it's these two identical twins, they're separated, birth by accident, they're adopted. She's adopted by him. His wife died, and she's adopted by this mom, and they sort of like end up finding each other by accident. But it's a sort of like nature versus nurture. Like they've grown apart, but how much does like knowing about like, and now when they find each other, they kind of like all just move in together, and just so that they can stay together during the high school and going onwards, and never let each other go. And how much is knowing this person's value of I think his name is Tamara and this is Tia. I might be getting it mixed up, I'm sorry. But how does knowing one of them help you know about the other? Like, what is the mutual information? Like if one of them ends up being really good at spelling, is the other one also really good at spelling, or softball, or so on? So it's like, how much does knowing a value of one variable tell us about the value of another? So that's like nature versus nurture. And if they're not really the same, like even in genetics, for instance, then that sort of shows how nurturing could be a difference, or how some things are also epigenetic, like changes in our environment, and other things that can change, that can add modifications to our DNA sequence as well. So basically, what I was trying to say is that we can use information theoretic algorithms like RTN that packages using mutual information to reconstruct transcriptional regulatory networks from gene expression data. And gene expression data can be of any form. Like you can have the genes, and you can have like time points, or maybe you want to have it like patient samples, for instance. Like you could have, instead of this one, you could have like samples of data instead of time points. So this could be samples, it could be individuals going across, like patient samples, or maybe cell lines, or just something that you're going to measure. Then, like you can see here that these are dendrogram clusters, and what those are trying to do is it's trying to show, okay, well, like we're clustering these genes out of these 6,000 based on the similarity using some hierarchical clustering. So that's what this is doing. It's shuffling these genes around. So you'll notice that here the expression patterns, like the black and green regions, they tend to be very, very similar, and those genes are then sorted like in that way, and so that the most similar genes are next to each other in terms of expression at different time points, because there must be something going on, similar things that are causing them to increase or decrease in expression. So we use this information for 18 different time points, for instance. So, pretend in this example that you're looking, as I was mentioning, you have gene expression data. So this is gene expression data for different individuals. So these are like the IDs, for instance, 7, 13, 21, 22, 23, 24 from a brain region of the brain. And what you're trying to do is looking at Alzheimer's disease stage to try to figure out like transcriptional regulatory networks, like which genes, like pretend like there are thousands and thousands of genes, for instance. So this example is probably not really that accurate or that good, but so usually in high dimensional data, you have like thousands and thousands of genes. You have way more genes, so this would be like in the thousands, for instance, or it could be much bigger. And you just usually have, so it usually looks more like you have more genes and fewer samples. So the genes and then samples is much smaller. So this is actually opposite in a way, but this is just an illustrative example that like just pretend like you have so many genes, thousands of genes, in our genome, and like APO is like a huge risk factor for late onset AD. APP, amyloid precursor protein, the PSEN1 is also like, so these two are really huge risk factor proteins. And then this is other gene, for instance, and then you have like the stages. So like, when somebody's control and they have a healthy brain, and then this is when there's mild Alzheimer's disease. So you can see there's already some neurodegeneration happening across the brain. Some neurons are dying. And then this is like severe Alzheimer's disease where people are already exhibiting symptoms of, forgetting other elements about themselves and their lives. So we have these gene expression for these control, initial, moderate, and severe. Maybe we want to learn some transcriptional regulatory network. Like what we would try to build is maybe, like if we know, like maybe let's say for instance, maybe this may or may not be accurate, like, but let's say that we believe that these could be transcription factors, for instance. Then like does A4 GALT, does it regulate, like let's just pretend, for instance, just for illustrative, this might not be biologically correct, but let's say like we're going to try to learn, does A4 GALT regulate like APO, for instance? That's what we're going to try to see, okay, what regulates what, is this regulating it, does this exist? That's what we can use mutual information for, like use this data here to try to infer something using mutual information. Or like, what is this mutual information that they share? The higher the mutual information, the greater the confidence there is in this edge existing. So as mutual information goes up, then they are sharing a lot with each other. And then this, basically this transcription factor has a lot of information that we can learn about for this gene. It's probably involved in a key aspect of regulation. Or maybe it could look like in this network that maybe also, let's say there's another gene, but like it's not visualized here, but out of the thousands, we also could have like, let's think of like REST as another gene. Like let's pretend it's in the list of thousands. And like does REST regulate it, so we're going to try to figure out, like, is REST also regulating this gene? And maybe it's also regulating like maybe it's regulating like the SEMA6D. So we're trying to learn all of this from the mutual information. So we're going to look basically across, we're going to scan across and see, okay, well, for across the different conditions, this is this value, this is A4 GALT, and we also have RESTs as well. And we're going to figure out the mutual information here between A4 GALT and APO. What is the mutual information that they're sharing? And when we calculate that, that's what this is telling us, essentially, like how much information. And the greater the mutual information is, the stronger the confidence is that there is an edge between them. And if we tell as an input that this is a transcription factor, then we'll know that the directionality will go in this direction. And if the APO could be a transcription factor, for instance, again, this may not be biologically correct at all, but then there'd be an arrow going in the opposite. Maybe that implies maybe co-regulation. But this is how we can construct using mutual information. So you can have information for the genes, and then here you have information for the Alzheimer's disease stages. And we can see here across time, for instance, and the individuals here are in different categories. So these people don't have it. These people in the initial stages, initial and moderate stages, and this is in severe stages. We have two different individuals. So this is like typically bulk RNA sequencing, where you have gene expression values and for a particular region of the body, like here it's a brain region, for different individuals, and they will express different phenotypes. Like these people will not express anything. They'll be the control healthy people. These people will start to express maybe just initial forgetting and some tangles and beta plaques in their brain. And these people will really have huge cognitive decline symptoms, and this also shows that it's a progressive disease. So I just sort of want to explain again like what I was talking about. So I was mentioning that here we're looking at the expression of 5 different genes in 8 different patient samples. So so the number of patient samples n is going to be equal to 8 in this example. And I was mentioning again that this is not really how high dimensional data looks, and in reality we have around like 20,000 different genes, in that region in our genome. And if we have 8 samples, so we typically have G, the number of genes, which is here. This is just an example. Like you can kind of see it. Maybe it's a little bit more visible what the genes look like. They're even more, and I can keep even zooming out even more to kind of show you that there's so many genes. There are like 20,000 different genes. And then if we have like 8 samples, for instance, then it looks really like this. So my point was just to show you that we're kind of looking at it a little bit distorted in this example with, 8 different conditions like control, initial, moderate, and severe, and just 5 genes. But that just like is a toy example. In reality, we have like so many genes, and it's the opposite shape where it looks like nice and stretched out like this. So that's what I was wanting to show you guys. And so this is just a toy example. So like this, we're looking at the monkeys on the tree, and Inspector Gadget is going to zoom out. And on this example, I'm sort of zooming out a bit more and showing you, okay, well, this is sort of like, these are what the trees really look like in the forest. And then here, if we zoom out even more, we can just see the whole thing. Like we can see more of that lake. What does everything really look like? And you have an even more perspective on how this is high dimensional data, because we have even more features, and the features are the genes. We have more features than we have actual labeled data, which is what stage you would be in. Typically we try to predict in machine learning, like what stage are you? Do you not have Alzheimer's? Are you in the control? Or do you have initial stage, moderate, or severe stage, or just in general, just do you have Alzheimer's, which is what these shadings are, and the green is that you don't have it. So in that case, what we would typically have is we have just a few samples, but a ton of genes. And I'm just trying to show you to help you understand that this is high dimensional data, because we have these features that are way greater than the number of samples. Even if you had 50 samples, the genes would be so much more. And that's also because of how expensive it is to do these types of analyses and get, this would be RNA sequencing data. That's bulk RNA sequencing, so that's also what this is called. I typically work a lot more with bulk RNA sequencing. I'm more familiar with that as well. So this is bulk. It's not as cutting edge as a single cell RNA-seq is, because that gives you more insight into the expression levels of different cell types. And we have so many different cell types. Even in our brain, we have oligodendrocytes, microglia, neurons, and astrocytes, to name a few. So this is bulk RNA sequencing, where you have the different stages for the patient, so their phenotypes, and these are average values in a particular region of the brain, for instance. And because we're studying Alzheimer's in this example, but it could be for the lung tissue, or they could be any tissue, pancreas tissue, or kidney tissue, any tissue, heart tissue. So again, just wanted to let you know that this is really what it looks like, but we're just looking at a higher level, overview of, with this toy example. But instead, we could also just look, instead of the different stages, for instance, just pretend we're looking at different time points for 5 genes. So this is an example we're going to try to work through. How will we find the mutual information and calculate it between these genes? So we want to calculate the mutual information between all pairs of genes, and then so here we're just going to pretend we really don't know who's the transcription factor, and we're just which ones aren't. So we're just going to calculate the mutual information between gene 1 and gene 2, gene 1 and gene 3, gene 1 and gene 4, gene 1 and gene 5, for instance. Then we want to calculate that information between gene 2 and gene 3, because remember that we already have it between gene 1 and gene 2. Then gene 2 and gene 4, gene 2 and gene 5. And then we also need to calculate the mutual information between gene 3 and gene 4, and gene 3 and gene 5. And lastly, we need to calculate it between gene 4 and gene 5. So the way that we can do that is by sort of looking within this given gene, and we can sort of do some binning first and say, okay, well, let's look at the expression of these different genes. Let's see how they are in terms of their expression levels throughout, these time points from 0 to 150. We can look at time points maybe for the same individual at different time points. Maybe like this guy, we can focus on his Alzheimer's disease progression. And I know these timings do not make sense, but pretend we're looking at 5 genes. We would be basically looking at all the genes, but we're just focusing on a small toy example here, where we typically, this data would actually look much inverted, where it would look like kind of like the opposite of how it looks. So I really hope that instead you can see that it would look just like this, like a rectangle with way more genes than samples or time points. But we're just trying to show here that we have these time points, and we're going to try to analyze based on these time points and the behavior. What is the information? Like, like maybe at a certain time point, as this guy's getting older, we would notice maybe that there is some neurodegeneration or some phenomenon is happening, as we're going down in terms of the time. So we're trying to basically figure out how can we group these genes, and that's how mutual information comes in. So we're going to figure out first within each gene, how can we bin it in terms of its expression levels, like, in terms of the minimum or the maximum values. Like, we should assign these values to given bins based on the distribution of each gene, because each gene has its own distribution, and we want to use that information when calculating the mutual information. So we're going to discuss the 2 different binning strategies that we could use.
And for all of these 5 different genes, for these 16 different time points in this example, we can sort of see what the typical gene expression levels are. So this is frequency, but what this really are, the y-axis, this is just mainly measuring like the gene expression values and frequency. So these are histograms. So this is known as a histogram. Each of these is a histogram. So what these histograms do is they come up with their way of sort of binning these observations into different bins over here. Like, so they're coming up with their own binning strategies based on their binning. And we can see gene 1's expression values, gene 2's expression values are here, gene 3's expression values, gene 4's expression values, and gene 5's expression values. So right off the bat, what you could see is that these go up to like 1, 1, and 1. So these are like, so these 3, I'll just have like a little, maybe I'll use this color. These 3 have pretty high expression values going up to 1, whereas gene 1 and gene 5, their expressions, like this one goes to 0.5 and this one does 0.25, so they like have relatively lower expression usually. So if we want to compare how gene 1's expression is changing, it's pretty low in general for gene 1 and for gene 3. So if we want to see how these expressions are changing during the 16 time points, it won't make sense to just compare, okay, well, gene 1 is this value and gene 3, gene 4. It doesn't make sense, right? Like, if you have somebody that you know who's really skilled, like let's say your parents or guardians are really good cooks, right? And you may or may not be, please don't take offense, but like let's say that somebody who's been cooking a lot longer, right? For instance, is really good at cooking. So if that person is learning a new recipe versus you, that person is going to probably make it like a lot better than you could, because they're already going to be super skilled at it. Like, one of the things I love doing is I love to knit, actually. So this is my yarn here, so I'm trying to make something as well. But if somebody who doesn't know like as much like knitting, or who doesn't know as much of you know, like let's say you don't know enough cooking or somebody else knows it, the idea is that they're going to pick up like a lot faster than you in the same amount of time. The person who's skilled is going to pick up a lot faster than the person who's not skilled. So somebody who's learning how to cook a new recipe versus somebody who's skilled at cooking, learning a new recipe, the person who's skilled is going to learn it faster, right? So it doesn't sometimes make sense to compare these values of gene expression, especially when they're on different scales, right? Like if we had to say, oh man, like how come you didn't learn this recipe and this other person who's super skilled learned it better than you, or they made the dish way better? No, it doesn't make sense that way. It makes sense to say, okay, where did you come from? You're not as skilled at cooking. Sorry, again, please don't take offense. This is just an example. You're not as skilled at it, right? Or like for me, for knitting, this is my grandma. She is my paternal grandma, she is super skilled at knitting, and she can knit something that would take me like weeks and weeks. She can probably knit it in a couple of days because she's super fast, you know, at knitting. In general, she makes very beautiful sweaters as well. Like she's super skilled. And pretty much the idea is that something that would take her a much shorter amount of time would take me a longer amount of time. So everything is all relative. So the key idea is that we want to make sure we're looking at like on a typical level. You know, like if this amount of time takes me this much time to make the same material when I'm knitting, or if I'm cooking, or if I'm doing something, it might take somebody who's more skilled less time to cook, or it might take them, like my grandma, less time to do the knitting. So we shouldn't be comparing the quantities per se or the quality per se, but we should be looking at like the changes in general. Like, are we noticing that like I'm doing better? So my idea is that we're not looking at the absolute quantities for these. We're trying to find ways to bin these values and then compare them, because gene 1's expression is never going to be higher than gene 3. Never going to reach gene 3's level of 1. It's never going to reach gene 4's level of 1 either, or gene 2's expression of 1, for instance, or even gene 5's expression of 0.5 here. That's just not like how its value varies. But we should see over time, are we observing the same trends over these 16 time points? That means like when gene 1's values is going up relative to its other values, are we noticing the same behavior for gene 3 or gene 4 or gene 5 or gene 2, such that knowing something about gene 1's expression can tell us something about the expression of the other genes? That's what mutual information is telling us. Like in sequence logos, does knowing something about like a position along the motif, do we know more that that base is likely to be a given base or not? Like basically, how much information do we have? And if we know that gene 1 and gene 3 tend to move in similar directions, right? Maybe at a certain time point, they really spike and then they go down. Maybe the cyclical behavior where they shoot up and then go down, shoot up and then go, shoot up and then go down, shoot up and then go down. Maybe it's cyclical, or maybe it's just increasing together. Maybe it's all decreasing. So it's like we want to compare how it's moving relative, right? So I can't compare myself with the experts in the world who are great at knitting, or who are great at cooking, you know, great at video games, or great at sketching, you know. Like if I made a star that looks just like this, I should be happy. Somebody else could draw a very beautiful star, right? But I'm getting better, which is nice, right? So the key idea is we want to bin these things into bins, like maybe using equal width, equal density, and we're binning it based on each gene's own values. And we're binning it based on its own values, and based on its own values, and based on its own values, respective values, and its own values. And we're seeing how does its behavior across the 16 time points, how does that behavior change? And are there similarities in the changes between these genes and, you know, these genes and these genes and these genes? So we're basically taking all pairwise, and then these genes, and then these genes, and then these genes, and then like this and this and this and this and this. You know, like hopefully try to capture as many pairwise as possible. But we'll be basically looking at, you know, 1, 2. comparison of those genes. Then we'll be comparing 1, 3 then 1, 4, like those genes, and then 1, 5, and then 2, 3, 2, 4, 2, 5, 3, 4, 3, 5, and last but not least 4, 5. And what this is this is basically like this is a gene I. I can just write maybe gene A and then gene B. And we're just looking at all these combinations and we're just trying to figure out, like, you know, that's what we're trying to do. And again, these are like the expression values, and our goal is to bin them. You want to put these into proper bins so that we can try to see how are they changing together based on their relative values. You know, we want to bin them based on their relative values using either equal width or equal density binning. Those are two main strategies to bin them based on their relative expressions. Put them into bins. That's our goal.
So here I kind of want to talk about, you know, feature engineering, you know, deep dive into encoding and binning techniques. So basically, when we're using like machine learning models, for instance, you know, we can do feature engineering, where we can do feature encoding, going from categorical to continuous encoding as well. Like one-hot encoding is very popular for neural networks as well. But pretty much in that kind of encoding, if you have multiple classes, like if you have, you know, classes, like 3 classes, for instance, you know, like in one-hot encoding, what you do is if you have like classes C1, C2, and C3, then the way that you would encode having class 1 would be 1, 0, 0. For class 2, you would say it's 0, 1, 0, and then class 3 would be 0, 0, 1, for instance. So that would be one-hot encoding. So there are multiple different encoding strategies that you can have. And here, what we're going to focus on is like going from continuous data all the way to categorical data through like this feature binning. And we're going to do like equal width binning and then frequency width binning. So equal width binning is also known as uniform binning. And density binning is also known as frequency width binning. So you're sort of, you know, like binning so that you have the same number of objects in each bin. And equal width binning is your binning so that each bin is the same size. So what this is telling us is like, you know, equal width binning again, and credit goes to Satyam Kumar as well for this slide here. And this algorithm for equal width binning is just dividing a continuous variable into several categories having bins or a range of the same width. So this is a way to discretize, to go from continuous data to categorical data. So you have your maximum value and you have your minimum value, and again, this is defined as our range. This part here is the range of the variable. This part here is the range. Min minus max, and x would be like the number of categories. So this x part here is the number of categories that we want here. That's what this x is. That's what this x is, and this little is sort of like kind of telling you how this equal width binning should be done so that you have roughly the same number of items in each. So you have roughly the same width in each bin. So equal width binning is making sure that each bin has the same width. So let's say that this whole thing here, from this point to this point, is like this whole interval is supposed to be, you know, like the min to the max. I want to like kind of divide it up into these chunks that are equal. So this is not exactly equal, but they should all be the same, you know, roughly. But you know, you want them to be equal chunks. So this means that it is inclusive, and So this is sort of like a formula, like min, max on the list, and that's how we do equal width binning. We just figure out, okay, well, roughly what would be the width, and that's sort of how we define these categories. And I'm going to kind of explain it more. So yeah, this is just another example again about equal width binning when you're given this list of these values for x. Okay, and we want to put them into 4 bins. Okay, so we want to put this value into 4 bins. So we want to discretize these values here into like age bins, for instance. So these are the values sorted from least to greatest. And what we want to do is we want to find these bins. So what we do is we find the maximum value here. The maximum value is going to be, so the maximum value here is going to be at 55. So that's our max value, 55 is our max. And our min value is going to be 10. So if you take 55 minus 10, that's 45. 45 divided by, 12, divided, so 55 minus 10 divided by 4 is roughly 12. So that's sort of like the formula that's being used here to figure out the width of each bin. And what you want you to observe is that each bin, they have the same width of 12, roughly 12, units. But the number of items in each bin may not be the same. It really depends. So here, there's 4 items in this highest bin, and then 2 in this, third bin. Like 1 here in the second bin, and I think 1, 2, 3, 4, 5 in this bin 1. So that's equal width binning, that each bin has the same width, but may not have the same number of items inside it. And one of the popular ones is actually histograms try to represent equal width binning. And then equal density binning is like equal frequency binning. So you're trying to make sure, and the garbage bin is trying to say like it's equal density binning. You're trying to make sure that you have roughly the same number of items inside each bin. So each bin has like the same density. That's what's important here. It might be off, give or take, but based on if it's an odd number of things you're putting, if it's not completely divisible. But here we're trying to figure out, okay, well, we have 12 ages. Our x has these 12 ages. So remember, this is our x here. Our x is here. This is our x, a random variable. It's got these values here. We're trying to discretize it into these bins. So yeah, so then this would be, these parts here would be all in like value 1. These would be value 2. And you notice the same number of elements would be in each one. This would be 3. And then this one here would be 4. So roughly, like, they have the same number of items because it works out the 12 divide. 12 is a number of items here, 12 divided by x, now we want 4 bins, number of categories, and this is giant x. X is this whole random variable of the ages. So this n here is going to be 12 items because there are 12 ages. And x is going to be 4 because we want 4 bins. So the frequency means we want to have 3 items in each bin. That's how this works. So that's like this frequency of 3. That's why we have 3 items here, 3 items, 3 items, 3 items. And then we can kind of compare them, like this is Python code kind of using this age. If you have that age variable, this sort of shows you how equal frequency binning works and how equal width binning works. So it's sort of the opposite idea that equal width binning, you may not always have the same density in each bin, but each bin is going to have the same width along. It's just like the same marks. You might not have the same, like it's the same way of binning information, like kind of putting continuous data into groups or categories, for work. So equal width, they just have the same width, but we may not have the same number of items inside each bin. But equal frequency, on the other hand, is saying that each bin has the same number of items. It's really trying to, if it can be divisible properly, it is supposed to typically have the same exact number of items inside it, but the widths of these intervals might not always be the same. As long as each one has, you just sequentially put each one in the bin, the next one in a bin, the next one in a bin, the next one in a bin, so that you have the same number of items. And this one is just like, okay, we have the bin max, we have the regions, we're just going to assign whatever comes in this region, like there, it just, everything has equal width. And this is Python code as well. Like if you want to kind of look at the comparisons of, these are 2 of the main binning strategies we're going to talk about today. So basically, now we have two different strategies that we can use to bin the values for each gene. So we're going to focus on each gene. For instance, like we have these 5 genes, and we're going to look at gene 1 in this example. And then we're going to look at gene 2, 3, and 4, and 5. And for each of these genes, we're going to look at all of its values for whatever it is. It could be a set of individuals in different stages of Alzheimer's, for instance, or a bunch of different time points, or anything. It's just, all that matters is that this thing is just either it's like different people, and we just want to find that the changes in these expressions along for these should be the same. They should follow the same patterns in general, because maybe this gene has a high expression level than this gene, for instance. So it just makes sense to say that relative to this expression, how is it changing in terms of its binning as we go throughout the time points? That's what we're interested in saying. How does this change in its overall expression, looking at its minimum to its maximum? And like we're putting it in bins, how does this behavior change during time, or during, as we are progressing from patients who have no Alzheimer's to those who have Alzheimer's that's very severe? So like these conditions, as we're going down, these represent the similar conditions for these people. For these time points and for these different genes, and we're trying to see if the behavior changes are the same, and then there could be some mechanism of transcription that could be taking place. But basically we're trying to calculate the mutual information, because if their behavior is going in a similar direction, where maybe these maximum, this is like maybe this is the maximum values are here, but it doesn't seem like here, it seems like this maximum value and this maximum, these are higher values, they're not quite moving in the same direction, for instance, there. So we would be saying, okay, like, so the one way to say is that, okay, this way we could look at it is we could look at the minimum values or the maximum values, and we could say, okay, well, it seems like 0.24, like these values seem like they're the higher ones. For instance, like these seem like the higher ones for this gene. And then for the other gene, maybe like what we see is like 0.92 is really high. It's got really high values. Is there any like kind of connection between these? Is there any kind of connection? Maybe I'll also just, for similarity, I'll also just, highlight like 0.83, even though it's a little bit off. There's a little bit more of a greater difference. So we can look at gene 2 and see, well, these maximums don't quite be, tend to move in the same direction, right? So like the expression levels are on very different altogether, different levels. Maybe it's just the way that gene is. So we want a fairer metric of comparison to say that, okay, out of all these expression levels for this gene, how is its behavior changing? Then for all of the expressions for this gene, how is its behavior changing during these time points? And then for this gene, how is its behavior changing, like the maximums and all of that? Like, how is it changing? And do they tend to change in similar ways? So that's what binning is going to tell us. For each column, we're going to put these maybe into like we're going to put these in probably like a maximum bin. For instance, like a maximum bin could put these in one bin together. And then it could put other values like the second tier in another set of bins, and then the third tier, and then we'll have one for the smallest one. That's what we're doing here. We're binning along each column. And there are two different ways for us to do this binning. One is equal width binning, where the bins will have the same width along, like maybe we want like, we look here and we divide it into equal set of bins, with the width are the same. Maybe we take the min minus the maximum, and then we find the number of bins we want, and each bin will have the same width. And that could be a strategy that we just focus on, like, they have the same width, and, this is another bin with the same exact width, for instance. Actually, it didn't quite look good, but the general idea is we could look at equal width binning or equal density binning. Equal density is saying that each bin should have the same number of genes in it. Like maybe we'll have the top 4, the next top 4, then the next top 4, then the smallest top 4, for instance. Oh, sorry, it would be like the top 4, then the second 4, then the third 4, for instance, and then maybe the bottom 4, like the minimum 4, for instance, like the lowest. There could be multiple ways to bin this data for each gene. It just matters that we do it in a consistent way. So we can either use equal width binning or equal density binning. And the important thing is that we figure out what is the optimal number of bins that we want to use, and what strategy do we want to try out. So now we're going to talk about equal density binning, or also known as equal frequency binning, and we're going to walk through an example of how we would do that for these 5 different genes that we are interested in at these different time points. Remember, instead of time points, it could be something else as well. So let's get started.
So you can sort of think of equal density binning, like for now we're focusing on gene 1 and we're binning it. So you can think of equal density as sort of trying to find the bin strategy that we would have each bin with the same number of elements inside it. So for instance, in bin 1, we want to have four genes inside it. 4 gene time points inside it, like so for 4 different time points. So this is the expression of gene 1 across these different time points. So in each bin, we want to have the same number of time points as much as possible. I mean, if you have odd numbers or some things don't work out, then we might have to make adjustments. But think of it like a traffic signal where there's like a police car and he's saying, oh, he or she's saying, we need 4 people to go each time. Dun, 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 dun. Okay, done. Because we have 16 time points, so if we have 4 bins, we will have 4, 4, 4, 4. Of course, if this is odd, if it doesn't, exactly work out, like if we have these 16 time points but we want 5 bins, then, it's not exactly going to be equal number of items in each bin. And that will really depend then on your implementation of mutual information, the binning strategy for equal density. But we want each bin to have the same number of elements inside it. So that's the goal of equal density binning. So in short, in equal density binning, usually has the same number of elements. That's the goal of equal density binning. So here we have 4 elements here, we have 4 elements here, we've got 4 elements here, and we've got 4 elements here, and we sort the gene 1 values from least to greatest. So that's what we're doing over here, and we can see that I just sorted it from the minimum value all the way down to the maximum value here, and the time points are all shuffled around based on that. So I first sorted them and I said, okay, these are all like the first 4, so this belongs to one bin. The next 4 belong to the other bin. These next 4 belong to bin 3. And these last 4 belong to bin 4. And again, based on the other situations, it might vary. So, so in my situation in particular, my implementation, this is how I've done it based on, like, let's say I want, like, so what, right now we were looking at 4 bins. So what we were just talking about was this situation here. This situation. We were just talking about 4 bins. Yeah, maybe I'll just leave for a second so you have this. So this is like what we were looking at right now. Is equal density binning results for gene 1 for different number of bins. So if we have 2 bins, 3, 4, 5, 6, 7, 8, and 9 bins. And again, right now, I had just shown how we would calculate it for 4 bins, making sure we have like the 4 here, 4 here, 4 here, and 4 here in each of these. So the thing that I did is I just sorted it from the smallest value, so this is the minimum, to the min, all the way to our maximum value. So you just have to sort it, that's what this is saying, sorted it from least to greatest. So least to greatest here, least to greatest. The minimum refers to these values here. Minimum, and this is the maximum value here. And then you just bin accordingly. So what this is saying is 2 bins, so you roughly have 8, 1, 2, 3, 4, 5, 6, 7, 8. So you can see 8 here. And then the last 8 here for 2 bins. So you're making sure you have You know, this is bin 1, this is bin 2. And then for 3, it sort of differed because of my implementation. So so just try these edge cases. Maybe don't focus as much on them. But you've got 1. It has 3 items in it. Bin 1 has these 3 smallest. Then bin 2 has the next 3. Then bin 3 has the next 3. Bin 4 has the next 3, and bin 5 has the top 4. You know, so that's how it was partitioned. And then you can see how they kind of change a bit for 6 bins. You know, so this is sort of overall, like the overall values are like this has 3 elements in it. 3, 2, 2, 3, and 3. And again, this had like the way that this was split was this is 3, 3, 3, 3, and 4, 8, 8. This is like 4 items here. You know, you sort them from least to greatest. Now later on, we're gonna like sort it back and then see where they all fall. And again, this is just for gene 1, so you'll have to do this. This is for equal density. You'll have to do this for the other methods too. So now we see here, this is 3, 2, 2, 2, 2, 2, 3. This is 2 here, 2, 2, items 2, 2, 2, and 2. So I'm just counting the number of items in each bin. Again, this is a little, not the greatest implementation idea for 3 bins, so I just don't want to worry too much about that. But pretty much, either this is 2, again, 2, this happened to be 1, 2, 2, 1. It also, again, depends 2, 2, and 2. So the reason you'll see this is because we have 16 items, 16. So again, we have 16 time points. What's that? Clock. 16 time points. And if we try to have like 9 bins, which is what this is, we're not going to have 16 over 9. You know, we're kind of short 2. So a couple of bins will take the hit, like this bin over here. Bin 3 and bin 6 are going to have only 1 item inside them because you have 16 and you have 9 bins and you have 16 time points, 16 total elements here. So you're not going to have enough for 2 in each. So this was exactly 2, but then now you have more bins. So now you're going to have like less, and then, you know, 16 bins, you have each one is its own time point. Each bin corresponds to its own time point over here and its own expression value. So that's sort of what I would say, like for rounding purposes as well. Maybe just keep all the decimal places, then and then sort them based on all these decimal places, then round them afterwards. So that's sort of like how for equal density binning, you take the minimum to the maximum. That's how this is looking overall. You're going from minimum all the way down to maximum and you're sorting it. That's what the sorting is. Sorting it from min to max. For all these bins, I just kept the sorting the same, and I just sort of made sure that based on I want to have 3 bins, so I'm going to sort them so that I have 3 bins, 1 to 3. I want 2 bins, so I have 1 and 2. I want 4 bins here, 1, 2, 3, and 4 are my bins. 5 bins, 1, 2, 3, 4, 5. These are the bins that they are assigned to. 6 bins, it goes all the way from 1 to 6. 7 bins, the bin numbers are going all the way from 1 to 7. 8 bins, 1 all the way to 8. 9 bins, 1 all the way to 9. So that's how you do equal density binning here. And then what I'm going to do is I'm just going to now sort them time values least to greatest. So now instead you'll see here that it's just going from 0 all the way to 150. That's what we're doing here. We're just sort of sorting them by time. Just a progression of time. and we're sorting these values, and what you'll see now, like this is 1 1 1 2 1 1. So this is just going to be how I'm going to like have this, you know, show up for these different number of bins. But in reality, we're just going to choose a number, so you can just see how the bins and the assignments of gene value differs. So we can think of the initial histogram and how that changes based on like where will the cutoffs be based on how many bins I want. So pretty much just to clarify again is that you can see that this is gene 1 expression, and we're focusing on gene 1, and we'll do the same process for all the other genes. And we just sort of see that based on the number of bins here, like here 1, 2, 3, 4, 5, and the histograms are using their own binning strategy. But based on the number of bins that we want, the data points will be assigned to their own respective bins. Like we to see this is that this is our bin 1, 2, 5, and this is like usually how equal width binning is done, because these are all like the equal width, and you can see that the density, the frequency is different. So this is not equal density, this is like equal width. So this is like 0.05, 0.05, 0.05, 0.05, 0.05, but so that's what we would get to with equal width next. But I just want you to see the idea that based on the number of bins that we have, so as we add or change this, we would get like very different results, you know. And typically like normal distributions look like and that is usually the result of like, you know, we are taking all of these values from the histogram, and that's typically, sorry for the bad sketch, but that's usually how a normal distribution looks. But in general, like based on the data, like this is just showing based on the number of bins that we want, how do these original data points for gene 1, how would they get sorted into the different bins, you know, and that's what these are. Like this again would be like, this would be bin 5, bin, and this would be like another, the other bin. So based on that, we'll be focused on 4 bins over here. So that it means that we're just going to focus on this value right here, 4 bins. So we're going to take these values here, and we're just going to work with these for now. So we're just going to work with these values. Four bins for gene 1. And I just wanted you to sort of see kind of what happens if we were to change these a bit.
So here, like, let's say that we're now looking at 4 bins, equal density binning. So for each of them, again, for each of these genes, we're going to do separate binning. So we're going to put them in each of their respective bins. And here, let's go ahead with 4 bins and assume that we're trying to put each of these genes you know, into bins 1, 2, 3, or 4 at each of these time points. So now we're going to look at gene 2 and focus on equal density binning for 4 bins. And I was showing before how we can vary the bins and how we can get different results based on that. So what we're going to do is we're going to sort the time points by gene expression value. So we have these 16 total time points here, and that corresponds to 4 different bins. So please assign the sorted values into the B bin such that each bin contains the same number of elements. So in equal density binning, we want them to have the same number of elements in each bin. That's the goal. So let's focus on gene 2 again, you know, with 4 bins. So the way, so this is like, you know, the Cars show, Lightning McQueen, you know, the Cars movie, you know, like it was a great movie. It is a great movie. So let's pretend. So the way I think about it is like, imagine you're trying to go on a roller coaster. I'm so scared of heights, so I probably don't want to go with that analogy. But like, you know, you have to, you know, it's a queue. You have to wait every few people is allowed in on the roller coaster. You know, every few people like is allowed. The roller coaster has so much capacity. So similarly, you know, the number of people in line you have to go based on that. But how do we do it for equal density binning? Well, to figure out, which people get to go at each time, or which bins each of these gene expression is at a certain time point. And remember, time can be one of the factors that we can do these binning on. It really depends on the nature of the data. It can be time values, it can be different stages, like I was showing you Alzheimer's stages, for instance. So what you do is you have this for each gene. So now we're focused on gene 2. So we're going to sort it from least to greatest gene expression value. So you can see here this 30, yellow is 0.21, no, 0.129, and the maximum value is over here. So you can see this whole spread. So we're going to focus on equal density binning for gene 2. So now we're over here. And now we're going to see, okay, well, let's focus on this first car. And this first car is going to be bin 1, which is level 1. So it's allowed to go. Then we focus on the second car over here. And that's, 0.1502. So this is the second value in line. And it's still allowed to go. So the first value is here, and the second one is also on this level. So each parking level will have a capacity of 4 cars. So next we also see that, car 3 is going 0.164307309. This is the third value over here, and it's also going to make it onto bin 1. So the traffic light is saying, okay, it's green, you can keep going. Imagine the traffic signal is allowing just every 4 cars to go in, on one parking level. So we have the first 2 over here, they've made it onto the same level, and this one is also going to be able to pass onto level 1. And then we have our last one here. And so now this value is also going to be, this car, this fourth value here is also going to be able to just make the cut and make it on the level. So now we have 4, we're at capacity now. So now this next car is going to come. It's going to be really disappointed. It's going to say, huh? You don't have a spot for me? Okay, well, there's no more room on this level. Oh gosh, it's not letting me through. So I led the first 4, and then it's like, no more allowed. It's like, oh man, now I have to wait for the next level to open up. Now I have to go on the next level. So problem solved. There's another level that's open. So this car is going to go right on to the level 2, the next bin number 2. And also just a friendly reminder is that, remember these values over here, they've already kind of been assigned to our bin 1 here. So now we are, have 2, this is going to be our second value on level 2, a second car, 0.0417, it's going to be here on the second level over here, and then similarly the third car is going to be able to enter in as well, which is 0.57, so it's also going to be allowed. And this last 1.6028 is just going to make the cut. We have 3 cars here. We can only have 4 on each level. So remember, because we had 16 values and 16 time points and 4 bins, so that was 16 over 4, which is 4 is the bin size. That's the bin size. That's the capacity that we have. So these 4 are going to be, all these 4 cars have made it into our bin 2. So these 4 have just finished making our bin 2. So now no other cars are going to come. So now when we go on to the next one at a time point 110, 0.608, and then remember what we care about is the sorting of these expression values here. So now this guy's going to come and he's going to be like, oh, there's no more space for me. No, you can't go. These other 4 are occupied. You got to go on the next level. Sorry. So this guy's going to have to go on the next level now. The stop, so every 4 are allowed, and then the stop sign comes and you can't go. So now the level has changed to the level 3. So now this one is going to be the start of bin 3. So it's now allowed to go on to bin 3. Similarly, now we also have this next car, which is 0.622, and it's going to also be allowed on the level. There's 1 car over here from the same bin 3 already over there, so we can allow the next car. And the next car is also going to go to bin 3, level 3 of the parking lot. And then 0.79 is also going to make it. The 3 other cars, so this guy's just going to make the cut. And then, as you can predict, then the last next car comes, this 0.825 is going to say, nope, you're not allowed. We have 4 already. End of this bin, end of this parking lot. We have to have a new level. So, so pretty much just to sum up, what we already have is that we saw that these 4 have already made it this would be our bin 3. These first 3 here were our bin 1. The first 4 were bin 1. Then these ones have made it to our bin 2. 40, this one. Yeah, these ones were our bin 2. Bin 2. And then now we are finished our bin 3. So now this car has to wait and go on to bin 4. So you can see we're at the last bin. So we were supposed to have bins 1, 2, 3, and 4. So these next 4 cars are going to make it on the last level, and then we're done. So all of these get assigned to bin 4. These last 4 cars make it onto bin 4. And you can see that this is the maximum value car over here. So I hope that this analogy helped, sort of how we sort, not by time, but by the gene expression value. And then the first 4 make it into one bin. They make it into one bin. Then these next ones make it into the next bin. Then these ones make it into the next bin. And then lastly, we have like this bin here. And sorry, this orange one should be including this. So that's how we can get our bins. We bin them based by our gene expression value, so that each of these levels has only 4 cars, because it happens to be because 16 divided by 4 bins is 4. But that's what, like, the bin size will depend on the number of points and the number of bins that you want. So if we only want 2 bins, we would have, like, these would be 1 bin, 8 values, and then these other 8 would be the second bin. So that's how we do equal density binning. So each bin, each level has the same number of elements, same number of cars inside it. And it doesn't matter what the intervals are, they each have the same number of elements inside them. So again, hopefully this should kind of highlight how you can go from a gene, like gene 2, and you sort it based on its expression values from least to greatest. So least value all the way to minimum to maximum. Again, so that's, min all the way to maximum gene expression value. Typically write in cursive, but so this is the minimum value. It's not as clean if I write it online, but it's all good though. So we typically sort the value from minimum to maximum. We bin it, these first 4, we just have like a divider that's going across like every 4, in a way, just saying, okay, every 4 have made 1 here, just divided by every 4, then the next 4, then the next 4, like it's all based on your bin sizing, so it's not like as neat. But then after you finish sorting them out like this way, then it's just like you sort again on the time. You sort it here on this column on the expression value, but now you're just going to sort on the time again, and this will be like what values you get for gene 2 here using equal density binning. You can see like these are back to back, so it goes from its maximum value at time 20 all the way down to its minimum value across these 16 time points, right back to back. So you don't jump from 4 to 1. So similarly, we can do the same thing for each column separately. So again, we have to take each all the gene expression values, please sort from least to greatest gene expression value here. So what we're going here is we see that this is the maximum value here, and this also happens to be the minimum. So similar to gene 2, they're like back to back. But now we sort it not on time, but on the gene expression value here. And then what we see is that we can do equal density binning. And then after we do equal density binning, we can then get 1, 2, 3, and 4. We just divided where the bottom 4 are, the 4 highest values are in bin 4. The next 4 make it into bin 3. Then the next ones make it into bin 2. Then the next ones make it into bin 1. The smallest 4 are in bin 1. From least to greatest. So think of it like, every 4 make it onto a parking level. You're part of it, part of it, part of it. And as soon as you're not part of this level. So it's like, you can think of it again like this. For this bin 1, you can think of, okay, well, you're allowed. Okay, so you're allowed. Okay, that's it. You're not allowed over here. A new parking thing is in order. These 4 are in 1. These next ones make it into 1, and then these last 4 make it into the other parking level. So that's how we do the binning. And it helps to sort them from least to greatest value, and then bin them, and then in the end you just sort again by time. So that's like a really quick strategy. And you can do the same thing for gene 4, where we see these values is sorted originally by time, from time 0 to 150. Then we sort by, minimum to maximum gene expression value. Then we place, 4 in each bin sequentially. These 4 make it into one bin, then these next 4 expression values make it. Then, the next 4 make it into bin 3, and the 4 highest make it into bin 4. And then so we sorted it on expression value to get it, and then we restored the original sorting, which was by on time. And this shows like how we have gotten the bins for each time point using the distribution of this gene 4 as well, using the condition that the same number of genes, gene time points should be in each bin, same number of cars on each parking lot, same number of people are allowed on the roller coaster ride. And of course, they might be off based on like, if you have an odd number or, if it's not completely divisible. Here, it just turned out we have 4 bins and, 16 time points or 4 bins with 4 elements each. So that worked out, but it may not always be the case. But for the most part, it depends on the algorithm you're implementing. So here, again, you're sorting it and, you're putting it again for gene 5, we're resorting it. And that also goes to show you that we sort each gene column for its expression values. And then, then we bin them again, each 4, the next 4 in the next bin, next 4 in the next bin, and the top 4 are in this last bin 4. And we had sorted it on expression, and this is us, sorting it again on time, restoring the balance. Happy clock. So, restoring it, and these are like how the bins go from time 0 to 150. So we've gotten our equal density binning. So we'll have like the 4, like these 4 ones are like scattered here, 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 as well down there. So this shows the equal density binning. So this is sort of an overview, and you can see that the behavior changes, for equal density binning from gene 1, 2, 3, 4, 5, as we go along for each of these different bins, each of these different genes. Like their, as I mentioned, their expression levels are different. Like this is going from 0.004 all the way to 0.249. This one's going from 0.212 to 0.93. So it's even much higher. The comparison between these two, the values, it's like this is almost, this is like much smaller, much smaller range. Like this is like really small, and the range of values is different. So we really can only, some genes, for instance, in the cell always need to be produced at a higher level. Maybe they're housekeeping genes. Maybe they're doing some things that are really important in the cell. So they'll always be at a higher level than some of these genes. Or maybe they're also specific to the tissue or the cell type, So that's why we really have to go on how these bins have changed. These bins are based on the distribution for that given gene. So we have to go on how do these bin values change? How do the distribution of gene values in these bins, how is it changing? That's what we're looking at. So we're looking within each, that's why we're doing the binning for each gene separately. We're sort of seeing how does gene 1's expression values, how do those change for each of these time points relative to what other expression values it's had? That's what bins 1, 2, 3, and 4 tell us. And over here, we're also doing the similar binning where we're also looking and seeing, okay, well, how does that change for gene 2 across its values from 0.12 to 0.939? Like we defined our bins, making sure that there are 4 in each bin. How does that change? What about for gene 3? What we see here is that like at time point 0, for instance, then, genes 2, 3, 4 are in the smallest values. Then gene 1 and 5 actually on the second value, like second bin, like with respect to what they typically are. So like we can see here that at time point 50, if we're comparing across, well, it looks like gene 2 is in one of its highest values that it's been, like it's in bin 4. But, and these other ones are in 2 as well, and this one's also like in 3. But for gene 5, it's one of the lowest values that it's had. Its lowest value is here at 1. It's in bin 1. But for the gene 2, it's actually in bin 4. So it's one of its highest values at this time point. So we're sort of comparing not these values with each other, but the binning, the changing in the distributions of the gene, of that gene's expression values across the time points. We're sort of seeing the comparisons between gene 1 and like gene 2 is changing relative to its other values, and gene 3 changing relative to its other values, and gene 4 changing relative to its other values, and gene 5 changing relative to its other values. So it's all relative to its other values. That's how what binning tells us. But this is equal density binning. The results would change for for like equal width binning. So these are ensuring we have 4 in each of these bins. And you can see like how the behavior is somewhat different. Like we can see maybe like genes 3 and 4 are a little bit more similar, like they are at 80, they're at their minimum, and then at 100, they're at their maximum. So maybe there's something going on, maybe they're like buddies, that may be moving more together, but we have to analyze more things as well. We already see that there are quite a few overlaps as well, where we tend to observe like this 2, 3, 2. They seem to be like similar, like a little bit similar, but not quite, like 1, 4, 4, 1, 4, 4. They have that initial like behavior going on as well. So we have to keep our eyes peeled. You gotta keep our eye out. Do we really observe that? And this is equal density binning results again, for these 5 genes. These are the values, minimum, maximum. Minimum is in yellow, maximum is in orange. And this is just a sort of a summary back to back to back, how we can see their behavior, changing across the time points for equal density binning. So each bin has the same number of values. Rough, but if it's not exactly like, divisible, they should have approximately the same number, give or take a couple, so usually the same number of elements in each bin. So that's how you would do that.
So I sort of told you guys about like equal density binning, and then along the way for gene expression for gene 1, we talked about how this is, in a way, this is equal width binning, because each of these is 0.05, 0.05, 0.05, 0.05, 0.05 width, and each of these is like 0.2. So pretty much it's like relative, and it's how we wanted to split up these bins. So again, like each of these is like going across. This is like each of these is 0.05. Then each of these is actually going to be 0.02, 0.2, sorry. And then same with these as well. Like this is also like 0.2 in width. Each of these. You see the same thing here and here as well. And then for here, like on the other hand, this is like 0.1. Each of these is 0.1. You can see these are 0.1. And lastly, like each of these is also 0.2. So these are like equal 0.2. So these are like, this is what's known as equal width binning. So what you'll notice here is the density. The density can also be measured here by the frequency. Oh, sorry, the frequency. That's like how we measure the the density. In a way, it could be measured here, but it's not equal. So this frequency, again, it represents like density. And then usually, like in statistics, we kind of can make that equal in a way, but it's not equal density, but these are equal width. So again, these histograms are actually an example of histograms are equal width for that given gene. And what you'll see here is that this is bin 1, 2, 3, 4, and this is bin 5, 1, 2, 3, 4, 5. just writing it here to remind you guys. Bin 2, 3, 4, 5. The default seems to be 5 bins. And 4, 5. You can visualize this in R as well, like what these look like. So the default, it appears under the histogram function, is that they appear to just be 5 bins. But you see that how we define these 5 bins, it all is super relative to, like, the width will be different. So all we have to do is we have to set like 5 bins, you know, in that situation, and then we're going to figure out, okay, well, like, what does it look like if we have 5 bins? Like, where are the cutoffs? So we can visualize it from here. Okay, well, it seems like bin 1 will be between 0.0, this minimum, and then 0.2, and then this. This is how R does the binning, but we're going to do our own type of binning where we start with the minimum value and then we go to the maximum, and then we divide it into those number of bins. So we're not going to quite start at 0, but so we're going to have slightly, like, not as neat, you know, like at 0.6 or like at 0.4 or like 0.3. We're not going to have such neat cutoffs. They're going to be like sort of starting at a minimum instead of starting at 0. But the key idea here is that it does some sort of a width binning where it says, okay, we're going to have these at some value here. Like, that's the idea of a histogram. And the density, the frequency density, the number of items in it, it's not going to be equal. Like here we have 5 items in this bin 2, but like 2 items in bins 1, 3, 4. Again, 5 items here. This is how 5 time points actually. Here 8, a lot of them, like in the have very low expression for gene 5, and just a little bit, like I think this could be 3. So we have, so the breakdown would be like 8. Then it seems like that's a 1, 1, 3, 3, 8 plus 1 plus 8 plus 1 plus is 9 plus 1 10 plus 3 13 16. So this is like the distribution of the 16 time points here. We have 8 in this expression, very low. 1 and 1 over here. We've got 3 here and here, so about 6 out of 16 are here, which is slightly higher. But we can see that they all have like different expression dynamic patterns here, and we're just trying to see how their behavior is changing. That's what we're trying to do. So in equal width binning, what's really important is here we're not starting all at 0 like here, so it's a tweak. We're tweaking what this histogram looks like. So first things first, we're going to find what is our minimum value, you know, our minimum value of 0.04. So we're looking here for our minimum value at 0.04, and that's what we find here. We basically go on and we scan for the minimum value here. And then we also look for our maximum value, which we have is this value here, 0.24. This is our maximum value, so we just scan through what this whole thing is. We scan through it, we find a minimum maximum value. Other ways we can just like kind of, you know, sort through this from minimum to maximum. So this is min, and then all the way down we're going towards our max. And our maximum value is this value here. That's, you know, right at the bottom. So then what we find is we take our maximum value here, then we subtract our minimum value, and when we take the difference between these points as we go down, that gives us our range, which will be, you know, equal to 0.2051. That's what this range value will be. And then the width of each bin is just the range over the number of bins that we want. So in this case, like it was 5, but again, it's just like it's not starting at 0, but our minimum value will start here, and then we're going to go in the width of each bin. So we're starting it here, and then this is how we find the width of each bin. We're basically going to a maximum, and we're dividing this region, this whole range here, which is 0.20521. That's what this difference is from here to here that we're traversing. We're going to divide that into a set number of bins. So the way to do it is to take this overall difference you want to achieve, which is the range, and then divide it by the number of bins. That's how we'll do it. We'll take this value here, and that's how we're going to do it. So here for this equal width binning, so we're wanting to have these 4 bins. So again, what we do is we can sort these values here. So right now they're sorted by time from 0 all the way to 150. So we can sort them by value from minimum to maximum value here. So we can sort them from minimum to maximum. So we get the minimum, which is 0.044616 for gene 1. And the maximum for gene 1, which is 0.249826, and we have our range, and this is a number of bins. So since we want 4 bins, we're going to just divide by 4, and we're going to have the width of each bin being 0.051302. So that is the width of each bin.
So what we can also see is that for the minimum value and the maximum values of like 0.044616 and the maximum of 0.249826, when we found these widths, those correspond for equal width binning with 4 bins for gene 1 to having this bin having this interval from here to here. And then these are the intervals from for bin 2, and the intervals for bin 3, and those for bin 4. And you can see that they all have the same width of 0.051302. So they have the same width. So it's equal width binning. So based on this minimum and this maximum value, we found out how to reconstruct these bins here for for equal width. And this is for gene 1, so we'll get like different values for different number, like gene number, like gene 2, gene 3, gene 4, gene 5.
So for equal width binning, you can kind of think of it as this idea for this gene 1. So like you can pretend that it's raining, and we have these 4 buckets, these 4 bins that have equal width, and here the equal width for gene 1. So for these 4 bins, we found that the equal width would mean that each bin has a width of 0.051302. This would be the equal width. So that's what's shown by the ruler over here. And so we just line up these 4 bins, and this is like Oscar the Grouch. You can pretend it's a Sesame Street analogy in a way. I grew up, I love Sesame Street, so I'm really a huge fan. I'm going to be a new aunt too, so that's exciting because I'm a first-time aunt, and I'm going to have a little one on the way, so we're going to enjoy watching one of these shows together, so that's so exciting. But in any case, imagine that it's it's raining outside, and these water droplets that are falling, and each of these bins is the same width. So maybe you can tell that this one's going to probably get 2, and this one might have 1, and this one just really seems like it's just going to have 1 droplet falling in, whereas this one could have 2 potentially. These could have 2. So it really depends where it's placed. So they have the equal width, and they could have, like, maybe one bucket can have more rainwater falling than others. It's just the luck of the draw. So that's what happens with equal width binning. You have these buckets at the same width, and it's just you place them out, and some buckets may end up with more water than others because maybe it's more concentration of rains falling there, the wind and all that stuff is also impacting things. So similarly, the expression might be that maybe based on the distribution of values, some buckets will have more values than others, for instance, and we can get some of that intuition by looking at the histogram for each of these genes. So for instance, what you might realize is that it seems that for the beginning width, like this is equal width, but they start at 0, which we're starting at the minimum value here instead. But what you can sort of tell is that a lot more values are concentrated in the later on values. So that's how this distribution looks. So this thing would be called a left skewed distribution. So an analogy one of my first staff professors had taught me is the idea of skewed left. Means that if you were skiing, if you're a little dude skiing, you would ski, yeah, that's you just ski to the left. So that's like, it's more like a left tail distribution. So that's a little smiley like skiing down. So it's a skewed left distribution because like it's more spread out here. There's like less density towards the left side, and it's more concentrated towards the right. So it's like a slope going down. So you'd be skiing in the left direction. So here you would expect in these values, so typically as we're going across from here to here, what we would expect is that the values, the gene expression value is increasing. So gene expression is increasing. Increases. So let's actually see what happens when we are doing this binning. So it's an increase in gene expression value as we're going from left to right. If we're going from here to here, there's an increase from left to right here. So let me tell you what these bin sizes exactly are.
So here for equal width binning, we're trying to create 4 bins for gene 1's expression. So here this ruler is measuring that, the equal width binning, and these are what these intervals correspond to, as we talked about before. So this is the bin for bin 1. It's the first bin. This is the interval for the second bin. And this is the interval for the third bin over here, and this is the one for the fourth bin. So for instance, the first bin is 0.04461, 6311, all the way to 0.0959188. And each bin has a width that's equal, that's 0.0513024889. And that's how we got it. This is Oscar the Grouch, it's an allusion to Sesame Street. So the idea is just like, rain falling. And we don't really know like which bin is going to fall into. There's wind, there's all these things that are happening. And we just want to see that they have the same, we're just having these buckets next to each other, and it's raining outside. These rain droplets are falling, and we don't know, this one over here is going to like fall here, or like, go here, or like go here, or no, we don't know. It's like just a rainstorm in a way, and we're just lining these buckets out. They have the same width. Don't know how much water we'll get in the end. That's kind of the idea of equal width binning. So for instance, now let's look at the first time point, time 0 for gene 1. So it's 0.176955. So time 0, we have this raindrop here. This is its value. Which bin will this gene expression value at time 0 fall into? Will it fall in bin 1? Will it fall in bin 2, bin 3, or bin 4? Which one? This is its value over here. So what we're going to do is we're going to try out each of these, and ultimately we found, bingo, we found that it's going to fall in bin 3, because 0.01769544813 falls in the interval from 0.147222129 all the way to 0.1985237. So it falls right in this interval. So this raindrop is going to land in bin 3. So that is the value that we're going to assign at time 0. So here, so this gene expression value at time 0 would fall into bin 3. So that's what over here we've assigned it to bin 3 for this time point. And now we have the other time points to try out, and we just have to see which of the values, which of these intervals they respectively fall in. That's the goal of equal width binning. So here we're going to also do equal width binning for the next one, which is time 10. So that's this value 0.08509. So we're going to see, okay, well, which bucket does it fall into at time 10? So which value is 0.085009 in between? I think it's going to be this one, bin 1. And indeed, it is bin 1 because this value is in between this interval, middle and high interval. So that's why this gets assigned to bin 1. So indeed, we put bin 1. So it's this value at this gene 1's expression at time 10, which is this value, is assigned into bin 1. So it's one of the lower expression values for this gene 1 in terms of all of its other expressions. So remember, all these bins are how we're going to bin it based relative to its own distribution. So this is equal width binning. And again, this is the equal width. This is the width of each of these bins here. Then again, we're going to look here at time 20. We're going to see, okay, well, where does this value fall into here? And we're going to say, okay, well, I think maybe it's probably going to be bin 2 because it's in between this, this value is in between these two values. So indeed, it's bin 2. So we're going to assign it to bin 2. So that raindrop falls in bucket 2. And that's why we assign it to bin 2. And similarly, we just go through each of these values here and we just find out, okay, well, which of these bins is it likely to fall into? Or will it fall into based on these intervals? These are these interval values. Like, for instance, this 0.185204 is going to fall in bin 3 because, 0.18 is in between 0.147 and 0.19. So that's how we can finish, doing all of the binning for gene 1 in terms of its expression values here. And these width binnings and these intervals, like minimum and maximum, are going to depend. So this minimum and this minimum value, this maximum value, it's going to really depend gene to gene. So again, you can sort of see that, based on that, we have 4 that fall in bin 1, 2 time points that fall in bin 2. We have 4 that fall in bin 3. We have 6, a lot of them that fall into bin 4 based on the width. So this is different from equal density binning where each of them would have had 4, 4, 4. That's sort of different. And now we can also focus on doing the same thing for gene 2 in terms of its equal width binning. So we're going to focus on this part here, equal width binning here, this part for gene 2. And now, as I mentioned, we're going to do each column, each gene separately. Its binning will be done separately. So again, we take the minimum value here for gene 2. We find the maximum value for gene 2. So we find the range is the difference between the maximum value and the minimum value. So this is the minimum value here. This is minimum value here. Maximum value here, all maximum and maximum. So this is the range. This range covered by the min to the max is going to be 0.809705. And what we do is we just divide it by 4 and we get the width of each bin. So here it's like sort of sorted by the time. Not by the time, but the gene expression values, and that sort of tells us how wide will gene 2's bins be, and you're going to see it's going to be different from what gene 1's width was. So here, based on this width and we just can derive what these bins will look like. So based on the width of 0.0202426, this will be the interval for bin 1, interval for bin 2, interval for bin 3, interval for bin 4. And I know, like, when it's equal to that value, those will be like boundary cases, and then it really depends on your implementation for how you want to incorporate them into the binning strategies. If it's equal to this value, do you want to have this, being greater, less than, or equal to, or just a strict less than? How do you want to do that? That sort of depends on your implementation for it. But that's kind of how we do this equal width binning here, and that's how we derive the intervals. So that means the last one is going to go from 0.73659 all the way up to 0.939016. And again, we can do the same thing for this gene 2. We have a updated width here, we have our updated intervals here, and we have these updated list of genes. And we're just saying, for instance, that for this value over here, 0.393809, it falls in bin 2 because it's going from 0.33 to 0.53, and this value falls in bin 2. Similarly, these maximum values will definitely fall in bin 4. The minimum value should definitely fall in bin 1. And then, for instance, like, let's see here that 0.608, it falls here in this interval from 0.53 to 0.736, so it's going to be in bin 3. So that can kind of make sense here. Then we can just sort again to just to see how many values we get. So we have like 3 values for bin 1. We have 3 values also for bin 2. We've got like 5 values for bin 3, and 5 values for bin 4. So it seems like we have a lot of values, like using this binning strategy. Seems like we have like a lot more strategy, like, you know, this, and then, like, you know, we have like higher for the other two bins, for instance. So it seems like it could be like skewed to the left, you know, the guy skiing to the left in a way. That's what this will tell us for this gene 2. And now we can focus on gene 3 for equal width binning as well. So we're also not just doing it for gene 3, and then we'll do gene 4 and then gene 5, and then we'll have all the respective bins for each of the genes. So again, for gene 3, so here is sorted by the time, but here you can see that I'm sort of sorted the minimum and the maximum values. And then, you know, minimum value of 0.030872, maximum value of, you know, this 0.908 value here, the same thing here. We can find our range will be, you know, going from the min to the max. This is covering 0.905. We divided by the 4 bins that we want, and this is the width of each bin. We do the same thing where we start with a minimum and then end with a maximum, and then we use that width of each bin to derive like each bin's threshold. So typically something falls in bin 1, it's going to then be between this interval, it's going to be between like 0.003087. It really depends on your implementation, and this is value for the gene expression, and it really depends on what you want. So this could be like 0.22946. This is what bin 1 would be. Like that would be the interval, and if it's up to you if you want to be less than, or, you know, this is a point, or if you want this to be less than or not, or just, you know, strict. It's really up to you how you want to handle that situation here. But that's sort of like how bin 1 would work for gene 3. And the same thing, you can see that the width is changing. And, you know, we update the width here of like 0.226372232. And again, we're just binning, like these are all updated using the previous slide. Like, remember, these are the intervals for bin 1. And, you know, the minimum as we expected is in bin 1, and the maximum is in bin 4. And for instance, this is in bin 4, this 0.8211, because it falls within this interval of, you know, 0.68 all the way to 0.908. You know, that's why 0.82 falls in that. So that's why it's assigned to bin 4. So we're just going through, we're just assigning each value, you know, based on, does it fall, you know, within these bounds? Does it fall within these bounds? Like the raindrop, wherever it's placed, you know, geospatially, does it for which bin it's going to fall into, you know? And we sort it again, like, you know, just we're sorting it by the bins, and we can see that it really, like this guy here is really happy because he gets a lot of values in bin 4. It gets like 6 values, gets a lot of values. This guy got 5, he's kind of happy as well, and she's like kind of a 2, okay, and it's like, I only got 3 raindrops, you know. Raindrops keep falling on my head. It's just like that same song, it's in my head too. But that's sort of how you do binning for gene 3. We also do it for gene 4 as well, the same exact idea, where, you know, we look here and you can see here I kind of put the result here, but you know, because it's the same idea, like we just look for the minimum value, we look for the maximum value, and then min and then max. We find our range, we find the width of each bin, and this is like the same idea for gene 4, like we find the width and the minimum and maximum values, and the width of each interval. So we can kind of just, like, you know, take this value plus this width equals this maximum value. These two are the same again. So I'm just going to explain again how we get this. So we know that we have a minimum value here, we have a width of each interval, so that's how we take both of these values to then find what the maximum value is. Then we also know that this value would carry over here, and again, it really depends on you if you want to have this value, like, you know, like be included in this interval or not. But like then this value carries over here. You use these 2 parts, 0.27997 plus this 0.224408 to give you this value, and then these 2 values are the same, you know, here, and then this plus the width gives you this value, and then these 2 are the same, this plus this width. So the difference again between these parts here is going to be the width. So this difference here is going to be, distance from here to here is going to be your 0.22440 8. That's what it's going to be. Between this interval, this is the minimum value and maximum value for each of the bin intervals. Bin 1, 2, 3, and 4. And that's how we get it, you know, like the minimum value plus this width of each, which should be exactly the same for each of the bins, will give you the interval for each bin. So the same idea, you know, we adjusted our equal width, and, you know, we put those values into our respective bins here. And you can see again, like, you know, so just to, you know, sanity check that this 1 corresponds to the minimum value here, and this maximum value here is corresponding to bin 4. And for instance, this 0.513 goes into bin 3 because it's 0.51 falls between 0.50 and 0.72. So that's why it goes into bin 3. This value over here goes into bin 3. So this kind of shows you again. So please let me know, guys, if you have any questions on how this works. Again, like 0.31096 is between 0.31. It comes between 0.27 and 0.504. So that's why this value gets assigned to bin 2. This 0.31096 gets assigned to bin 2. And you can see again here that, you know, we have a lot of values in bin 4. We have these 7 values get assigned here. So here I'm just sort of wanting you to just see how it works, and that for equal width binning, you may not get the same number of items inside each bin. It really sort of depends, you know, where the water falls, where the raindrops fall, where it all lands up, you know, based on the distribution. All you're doing is just making sure that they all have the same width, you know, same exact buckets. It just depends on where you place them. I really hope you guys like my analogies. I know they can be like hit or miss sometimes, but hopefully, you know, I love telling like analogies and stories as well. And lastly, this is gene 5. So we're doing equal width binning, and we're focusing on its minimum and maximum values and then finding that range. As usual here, we can find this minimum value here. That's what this part is. This minimum value here is going to be this min here. And that's also the same minimum here. The maximum value here is going to be this value here is going to be the max. And the range is going to be this nice value here of 0.43, 41. That's what this, that's what this value is going to be here. That's this range. We plug this range in over here, and we get the width of each interval in each of our bins, 0.1085 here. And that's what we use here, 0.1085. That's going to be the width that we're going to find. And these are the intervals for each of our bins for gene 5. So again, sort of just reiterating, you know, you take your minimum value, you have your width, and that gives you this maximum value. That's sort of just reiterating, you have your minimum value here. That's what this part here is representing. Oh, sorry, this part here represents a minimum value. Then we have our width, which is what this column is representing, the width. And that gives you the maximum value, which will be here. So that's how we get it for each of these bins. And again, we updated our equal width binning. You know, this is the length, the width of each bin. And this is sort of showing how our binning works out for gene 5. You know, these intervals have been updated as well. And again, you'll see that the minimum falls in bin 1, maximum falls in bin 4. This seems like this is the only value that falls in bin 3 is at time 0.90 plus 0.273. It's pretty lonely, right? It's, you know, so again, this value for this, this time 0.90, you know, it's only when it falls in bin 3 because 0.273 falls between 0.219 and 0.327. So that's why it falls in bin 3. It's the only one value. And same with actually bin 2. It only has one value here. Seems like a lot in bin 1. Don't know why this bin 1 is still unhappy. And then, you know, a little bit fewer, but, you know, still quite a bit in bin 4. So, you know, when you combine them all for equal width binning, we can sort of see again, similarly, the behavior of the binning, you know, based on these gene expression values and the equal width binnings. And they're all relative to the expression for each of these genes. So we think of it like sort of like how a histogram works. Only your min and is not like really at 0. They sort of like defined based on the minimum value here, but you can also probably tweak the histogram as well. But it's a similar idea of how a histogram works. And you can see how these behaviors are changing. Whereas, for instance, here, if we look at time point 10 for gene 1 and 2, and also 5, it's actually like the lowest value for gene 1, 5, and 2. It's, you know, for gene 2, it's 1. Gene 1 is also 1. For gene 5, it's also 1. But for genes 3 and 4, it's actually like in 4. So it's the highest value for genes 3 and 4. It's actually like one of the lower values for genes 1, 2, and 5. And hopefully over here, what you can sort of see is that we, you know, just sort of, I was just mapping them between the two so you can have a better idea for how this looks, the comparison of gene expression values for all 5 of these genes here, how equal width binning makes them look here. And again, we were observing some interesting behavior, I would say, when it comes to like this gene here, these tend to be like a lower value for the time point 0. So this is just an example, like when you're given this data, like how would you tend to interpret, you know, what's going on over here, you know. So that's what we're going to try to do, is see relative to their expressions. These are their bindings relative to their own expressions. You know, for these equal width bindings, we found their minimum value for each gene. Their maximum value is maximum value. Then like the number of bins, which we held constant for all of these as 4. So all of these bin values have to be between you know, 1 and 4, 1, 2, 3, or 4, and these are all discrete numbers. So this is an example of us going from continuous data all the way to discrete. Is discretizing it based on the number of bins. And sometimes, like, you know, in some programs you can write it where, you know, you can slide the bins and you'll see how the histogram changes in its shape as well. So this shows how you're discretizing, so making it discrete, something countable, going from these numbers with these decimal places, like, you know, on pi you could go like 3.1412 like forever and ever. You're putting them into these discrete bins here, and that, so this is called discretization again, from continuous to categorical. But we're looking within each gene to get its binning. So the binnings are not like universal, like the widths were not universal. It was each gene, you know, each gene's minimum value, each gene's maximum value, then defining that interval and then binning it there. For gene 2, the same thing, it's min and max, you know, defining the width of each bin, and then kind of sorting them. So we did it for each of these, and we're going to then compare their behaviors relative to the other values. And that's what this is telling us, that these 4 are telling us that here for this gene 1, this is like one of the higher values it typically is. One is saying it's one of its lowest values. And that's what we're seeing here. We're trying to understand, like, it's a similar behavior between, you know, the highs and the lows, or anything that knowing information about one gene can tell us information about the other. So that's what we see here with equal width binning, and now with equal density binning for the same data here, we could land up with different results for how they look. You know, we see here maybe the lowest ones tend to be the same here, like this 1 and 1 tend to be here. These tend to be like the same, 1, 1, 4, 1, 1. This tends to be conserved, right? This one actually tends to be conserved, that same row, but like this 3 is a 2 here. This 2 is a 1 here, but these 1s are conserved here. So it's really different based on the binning strategy that we use. And I just want you to take a few moments to just please look here and see if you find like anything that's, you know, stands out to you. This is a 2 here, this is like a 3 here. Some similarities, some differences, but this is how we chose our binning strategy, and that's how we got our results. And there's no right or wrong answer. That's why there's so many same ways in machine learning and data science to get different results. Even with the same set of data, you can be very creative, get very different results. So what really matters is that you're taking some thought into why you're doing a certain binning strategy, and maybe seeing how sensitive are your results to like how you're binning them. So I mentioned that there are like these 2 binning strategies, equal width binning, equal density binning. So for the sake of this lecture, we're just going to choose one of them. I'll show you the results from equal density at the end, and then I can create additional videos to work through more examples of how we calculate mutual information. But let's say that we're using equal width binning with 4 bins, and let's just work with this example. So basically each of these bins, you know, in each gene, each of them will have the same width. In gene 1, for gene 2, they'll have the same width. Each of these bins may have different number of, you know, range ops that fall and made different number of items in each bin. But for each gene, each bin corresponds to the same width. Same with gene 4 and gene 5 as well. And we saw like they have different widths. So here, please write out all pairwise combinations of bin results for any two genes. So please remember that we chose 4 bins and we were like comparing equal frequency with equal width binning, but we chose 4 bins. So that means like for gene A, it can have values 1, 2, 3, or 4 for the bins. And then for gene B, it can be in bins 1, 2, 3, and 4 as well. So then the combinations could be bin, a gene could be at a time point could be in bin 1 for A and 1 for B, or maybe 1 for A and 2 for B, 1 for A and 3 for B. So this sort of shows that for a given time point, what are all the possible combinations that you could have for where a gene is binned in gene A and where it's binned in gene B. So that's what it represents. So please note that this is also going to allow us to analyze the behavior of both genes A and B in terms of the binning, like how do they move respectively, because remember, like this binning from 1 to 2, 3, 4, this is representing gene A's values increasing from 1 all the way down to 4, and this represents that's what it represents, and this 1, 2, 3, and 4 for gene B represents its changing with respect to its distribution. And for instance, this would show that they both have the same high value at this time point, like respective, like it's in the highest category for gene A and the highest category for gene B over here at this time point, for instance. But what this would represent, what this combination would represent, is that they both are on the smallest. But something like this would say that at this time point, we find the value is it's like one of the lowest for gene A, but it's among one of the highest for gene B. And this show that like 2 and 3 kind of show that, well, okay, maybe it's a little bit on the lower end for gene A and a little bit on the higher end for gene B at this time point. The expression level for gene A, it's second out of the 4th bin, but for gene B, it's a little bit on the higher end. So we're trying to analyze, like what's the breakdown? Do we tend to see that they have the same behavior together? And if they do, maybe knowing something about one of these genes can then give us more information about the other gene. So that's what mutual information is trying to tell us. So here, what we're going to do, I'm going to walk through a couple of examples, for the equal width binning. If you want to find the total counts for genes 4 and 5 now. So what we're going to do is, here I just wanted to sort of make it a little bit more structured the way I organize it. So these are the time points, from 0 to 150. But I just wanted to organize it so that I have like, gene 1 having its values, then for bin 1, then its values for gene bin 2, and then bin 3 and 4, and then I just wanted to just make it a little bit more organized so that to kind of face the same skeleton here of like one gene 4 being in bin 1, bin 2, bin 3, and then bin 4, and then gene 5 being in bin 1, 2, 3, 4, like just kind of making it like a little bit easier to see the pairwise combinations for mapping. So that's why I kind of reorganize this a bit. It just depends. You just have to fill out this table of all possible bin combinations for gene 1 and gene 5. So there should be 16 total rows here because we would have 16 combinations. So gene 4 could be in bins 1, 2, 3, and 4, and gene 5 could be in bins 1, 2, 3, and 4 as well. And these are the combinations for a given time point that gene 4 could be in bin 1, and gene 5 could also be in bin 1, or gene 4 could be in bin 1, and gene 5 could be in bin 2 with respect to its distribution of other values. Or we could have like an extreme where maybe like gene 4 is having its lowest value at that time point, and gene 5 is having its highest value at that time point. So it's in bin 4, and then this has been bin 1. So we just want to organize it this way. And what number of times does is I'm looking inside this data. So I'm going to actually end up using this part over here. I'm just using this here, this table here, to just get me all those counts that I want. And that's what this is telling me, that I'm looking in and how many times do I find 1, 1. So that's why I kind of reorganize it just so that it makes it a little bit easier to map, and I'm not like looking all over the place, it just makes it a little bit easier to map it. So 1 and 1, we have one time that this appears. We don't find 1 and 2 anywhere. 1 and 3, it happens one time. 2 and 1, one time. These other ones don't appear. 2 and 4. Okay, so what we see for 2 and 4 is that happens this time is one of them, 2 and 3. That's why I marked this as being 3 here, because I found it 3 times. And similarly, like 3 and 1 happens, 3 and 4 happens 1 time as well. These other ones are not found. It's 4 and 1 happens 5 times. That much. What I find here is that those happen 5 times. 4 and 1 happens here. 1, 2, 3, 4, and 5. So that's why this is 5 over here. And then we have like 1 of 4 and 2, and then 1 of 4 and 4. So that's how we have it. And then for this situation, we're using a pseudo count of 0.1. So that means I'm adding 0.1 to these number of times because some of these are 0, and it's a form of Laplace smoothing just to make sure that I still have some non-zero counts. You know, we don't really want a probability of like 0 because there's still some chance. We just want to just bump up things a little bit. So what we're going to do is just like add some form of smoothing, which is a really small pseudo count of 0.1. So we just add them together. So 1 plus 0.1 is 1.1. So we just add like the pseudo counts to these total counts. And then what we get is we get our total counts for genes 4 and 5. And we can do the same thing also for genes 2 and 5. So you just focus on those columns for gene 2 and then gene 5. And you know, that's what these values are. And then you just focus on the number of times that you find them here. So that's a gene, this is gene 2, this is gene 5. And you just focus on the number of times that you find the given combinations. And again, this just shows me the original one here. This is showing me the original one. This is me sorting it so that it looks as close to this as possible. It just makes it easier for comparison. So that's what this part here is going to say. I'm just focusing here. I'm using a pseudo count again of 0.1 afterwards. So then I'm going to say, okay, well, let's go from the top. Let's take from the top, you know, 1, 1, 1, 1, 1, 1. Okay, so I see that happened 3 times. Do I see any 1 and 2? 1 for gene 2 bin 1 and a bin 2 for gene 5? No, I don't observe. The next thing I find is you know, bin 2 for gene 2 and bin 1 for gene 5, and that's why I observed that actually 3 times. So I'm going to put a 3 here. So that's sort of how we go through. We're just counting the number of times we're observing these combinations here, and then we add a pseudo count of 0.1. And so 3 plus 0.1 is 3.1. And if you notice that this should sum up to the number of time points of 16. The pseudo count, you know, 16 times 0.1 should be 1.6 because we're adding a pseudo count for each count. Even if it has a count, we're adding the pseudo count to everything. So 16 plus 1.6 should be 17.6. So these are some sanity checks. Just make sure you are adding them properly. So this is out of all the 16 time points, how do they typically behave? That's what this total counts will tell you. And then lastly, we can also just do it for genes 2 and 3 as well. I just want to give you another example, just so you could kind of see that this is gene 2, gene 3 is here. And again, just to make it a little bit simpler, just to clarify, again, this is our gene 2. So actually, let me make it in green. So this is our gene 2. And then this part over here is our gene 3. I got cut off a bit, gene 3. And then what we're doing is we're using a pseudo count of 0.1. This 0.1 is the same thing that is going to map this 0.1 here. Is the same thing that maps, you know, this 0.1 is what these values are as a pseudo count here. And again, the same idea. Now, you know, I just sorted it again to make it easier. We don't have any 1, 1s. We see that we start from 1, 2. So that's why it's 1, 1, 3. It's 1. So that's what, just to clarify, maybe I'll just make it a little bit easier to also visualize it or understand it a bit. That we see like for 3, 3, what do we find here is we have these 2 points for 3, 3. That's why if we look here, we have a 2. Because we found 2 points, so there are 2 of these. So again, that's why there are these 2 points here, and that maps right here, and that's how we observe a 2 over there. So that just kind of is how we go ahead. We find 4, 4 as like these 3 over here. 3 times we find 4, 4, so that's why we have that. And again, these sum to 16, so that's great. The pseudo count sum to 16 times 0.1. 16 of these values, you know, 0.1, 16 times is 1.6, so it adds to 17.6. That's awesome. So this just shows how you can find the total counts for gene 2 and 3 combinations. So there are all these, you know, we have these 5 genes, and it really will depend on the number of genes you will have. But for these, we have the gene 1 and 2, 1 and 3, 1 and 4, 1 and 5, 2 and 3, 2 and 4, 2 and 5, 3 and 4, 3 and 5, 4 and 5. So these are all the pairwise combinations. I've just shown you how we can do it for genes 2 and 3, and genes 2 and 5, and genes 4 and 5, so that these 10 here. But you also can work on the other ones, and I'll try to put up some more videos where I work through these as well if you need more help there. So this is just kind of showing you a comparison of the total counts that we get for gene 2 and 5, gene 4 and 5, gene 2 and 3. What are these total counts looking like over here? And we could see that, well, it seems like gene 2 and 5 actually have a lot at 1 and 1, and these are actually the same. This is kind of what we would like to see, is that they have like typically similar expression patterns, you know, for the most part. But, you know, 1 and 1 is like 3.1. That's great. And we also see that 3 and 3 is also like 1.1. Oh, sorry, I'm like bad at selecting. Let me just put this. So 3 and 3 is 1.1. And this seems to be like a little bit weaker over here. Seems to be a bit stronger. So we can take this information as well. From this table itself, we can get the mutual information. But I'll kind of walk through it with you, you know, find mutual information between genes 2 and 5 and 2 and 3. But remember, they're all those pairwise values you have to find. So now let's see, okay, well, what is the mutual information between gene 2 and 3? Well, what would it be? So we can use the same table that we were just derived. So what we found is we sorted it like, you know, 1, 2, 3, and 4. These are all the values with the pseudo counts and everything. And now what I want to show you here is that we just sum up these values here. If we sum up the values above here, we're going to get, like, above here will be 3.4. These values are summed to 3.4. These next 3 values will be summed to 3.4 as well. And this next here, which is also just, you know, written as, maybe I should just have it like this. Oh gosh, like this, a little bit like that, yeah. So then these values when summed up will be 3.4. Then these values here when summed up will be 5.4. Then these values when summed up will be 5.4. So these would be the counts for gene 1. So what we're observing is, okay, for for gene 1, like what are the total number of times that we find it in at all in this you know, bin 1 out of all these counts. So just kind of taking the behavior, and we can find it in bin 1 even if gene 3 is in bin 1, 2, 3, or 4. All we really care about is when and where we observe gene 2 to be in bin 1. That's why we sum these values. So this is saying gene 2 is in bin 1 and gene 3 is in bin 1. That's what this first row is saying. The next one is saying gene 2 is in bin 1 and gene 3 is in bin 2. That's what we have here. This next one is saying gene 2 is in bin 1, gene 3 is in bin 3, and that's what the 1.1 is telling us. And lastly, gene 2 is in bin 1 and gene 3 is in bin 4. That's what this 1.1 is telling us. And our main focus is like, we don't really care what gene 3 is. We just want to find like the counts when gene 2 is in bin 1. And that's why we sum these and we get this 3.4. And this value maps from here to here. This is what this 3.4 is here. Okay, so you see that that's the same value, this here. And similarly here, all we care about is gene 2 being in bin 2. So I think it's just slightly a little bit messy. So you see that this is just like, you know, that's why we take this 3.4, because we are just holding on to gene 2 being in bin 2. And we're not really caring as much about the specific value yet for gene 3. We'll worry about that when we are taking gene 3's count. But for now, we're just looking at gene 2 being in bin 2 and all of the counts for any possible value of gene 3, 1, 2, 3, or 4. And we sum those values up and we get 3.4, which is here. And similarly here, we're just summing up these values here to get 5.4. And this is like gene 2 being in bin 3 and gene 3 being in bin 1. Gene 2 being in bin 3 and gene 3 being in bin 2, or gene 3 can be in bin 3, or it can be in bin 4. We don't care what exact value gene 3 has. We just care right now specifically on having gene 2 be in bin 3. And when it's in bin 3, like, we can observe any of these outcomes. And we'd use Laplace smoothing just so that we have, like, you know, this wouldn't be 0, for instance. And so again, lastly, like we get our 5.4 here for gene 2 being in bin 4 by just focusing on whenever gene 2 is in bin 4, regardless of what gene 3 is, it could be in 1, 2, 3, and 4, we just add that. So we get our value of 17.6 here, which it should be, and that's also another sanity check. So this is the total counts for gene 2 being in any of these bins. And similarly for gene 3, we do the same thing, and that's why I kind of like sorted it this way to make it a little bit easier. So you can sort of see the, like in here for gene 2, we can see the intensity increasing as gene 2's binning is going from 1, 2, 3, and 4. I did it that way. And here, when we're focused on gene 3, I've kind of binned it by, you can see that instead of being sorted by gene 2, I've sorted it by gene 3's bins. And this is showing we're going down gene 3's binnings from 1 to 4. And here we're focused on, for this part here, we're focused on gene 3 having a particular bin. So for it to be in bin 1, we don't care what bin gene 2 is in, so we just add up all these values here. We get our 3.4. That's what this is. We get 2.4 for the next one, because what we are concerned about is this part here. So for this 2.4, we're just looking here. Okay, well, let's just see any time that gene 3 is in bin 2. And then we sum it up, these values, we sum them up and we get 2.4. This value here is what this value here is. So that they are the same. That's where we get that value from. And similarly for the 5.4, we just focus on gene 3 being in bin 3, and we don't care about what bin gene 2 is in. It's just we're summing across all possible values for gene 2. And then you know, for the total counts, and then we get 5.4, which is what this is. And lastly, the 6.4 is we're just you know, holding constant, you know, gene 3 being in bin 4, and then summing up all the possible other values for gene 2 in 1, 2, 3, and 4, and we get 6.4, you know, once we sum up these values. So then we basically get these counts for the total counts for gene 2 being in bin 1, 2, 3, and 4, and here it's for gene 3 being in 1, 2, 3, and 4. So now we have this information here, and we want to now like find the mutual information between groups 2 and 3. And in reality, you just needed this, but I'm just kind of breaking it apart to make it a little bit easier to see what you know, what's going on. And from the same tables that we had before, I'm just adding some more that, you know, all these total counts are out of 17.6 here. So for the probability that gene 2 is in that bin, I just want to divide these total counts by, you know, for each bin by the total count. So 3.4 over 17.6 gives us the probability that gene 2 is in, you know, bin 1. And then the same thing for, you know, bin 2. Bin 3 is like 5.4 over this value. Bin 4 is 5.4 again over this value. And then they should sum to 100. Same with gene 3 as well. I just take each of these counts and I divide it by our total of 17.6 to just get these as probability values. I also do the same thing over here for each of the combinations. So this is like where you do care. Gene 2 does care about what gene 3 is here. That does count as 0.1. So these counts like are just holding gene 2's bin constant and varying across the gene 3's bins. And that's where we got these sums from. And here for gene 3, we're holding gene 3's bin constant for each of these rows and varying across the gene 2's bins from 1, 2, 3, and 4. And that's where we got these values from. You know, so it's all based on this, you know, like we had gotten these values by just, you know, holding this gene 2 bin constant here and varying these and summing these up. And that's how we had gotten this 3.4 to begin with, right? Remember, like how they map that, you know, when we were focused on like this part here, like how we found that is we just focused on this part, you know, when gene 2 is in 1, and we just take all these values here, we sum them up, we got our 3.4. This we sum them up to 3.4, and that's how we got our values for this. And then we did the same thing for the 2, we summed those values, got 3.4. So that's how we did it. But now we're also going to find for these combinations, our ability of combination for genes 2 and 3, you know, being in a certain bin. This is bin 1 and bin 1, 0.1, which is 0.57 once we divided out of our total counts. And remember, we had the Laplace smoothing, so none of these should be 0. And these should all add up to 100% because that's what we typically observe, what behavior we're observing for the binning for gene 2 and gene 3. And then to calculate the mutual information, now we're really getting here to the meat of the problem. We have your, how much knowing the value of one variable tells us about the value of the other. So this, you can think of this part as being, I sort of say that this. So what you can see here is that this M here, I just defined it here. This is our M. Our C is going to be gene 3. We have this logarithm. And then this is our PM, comma C is the same thing as this. This part, they're all the same. This part is the same thing here. This part here is just going to be this part here. That's what that's going to be, and also the same part over here. That's what this is going to measure here. Then the next part is just kind of like, this P of M, which let me have this as a light green here. This part here is going to be this part over here. Then our P of C is, let me just kind of have this in orange, just, it's a lighter color. This is a C over here. And this over here, that's what they're going to map to. And then we're pretty much just going to take our M. This is our gene 2. This is a probability, like, for that. These are our bins. The probability of the gene being in each of those bins that we had found for gene 2. So we see that all these ones should be the same. Here 19.32, and then this is also the same here. So they're moving the same direction with the bins that we're in, that we had just found. This is for a P of C for each of these different bins as well. Like we just kind of, probability that gene 3 is in bin 1, 2, 3, and 4. So you can sort of see that the way that this moves is that each of these, if you observe a little bit more closely, what you'll find is that this is one cycle, this is another, this is like another cycle here of 4, 2, 3, 4. This is another cycle of 4, and this is another cycle of 4. So they're all like the same values. Like that's how I did it for this. These are all the same. These are all the same again because of, that's how this is moving in this way, this way, this way, this way. So we have these probability values. So now kind of like, we're just kind of combining everything now to find this mutual information between genes 2 and 3. So you can see here, it's the same thing. I've just kind of tacked on a few columns. So what this part here is this part is the denominator of this mutual information formula here, where that's what is measuring PM times PC. So we take each of these elements of PM and PC and we multiply them together, and then we get this expression here, which is the same expression that's going to be part of the denominator here and here. Then for this probability that you see here, P of M and C, P of M and C, that's just equal to this, joint probability of them being together, which is here and here, here and here. And that's what this part here is, and that's what this part here is as well. And this yellow part here is just here. So you can see that this part here is all what this yellow part is looking at is this. Then this part here, the orange part that we're using, the P of M and C, is just given by this. And that's what these values are and these values are. So we just can take these values, divide them by these, and get these values. And 6.25 divided by 2.63 is equal to 237.25, like in percentages. So that's how we get this. So this part divided by this part will give you this part here, these fractions. Then you take this fraction and you apply the log base 2 on it. So remember, it's a base 2. So you just have to apply the log 2 transform onto it. And then you're going to get, like, just take a log 2 transform, and that's what this is saying, and you get these respective set of values. Then you just have to multiply this value here, which is this part here. That's what this part is mapped to. You just have to multiply this value, which is again what this part here is. You just have to multiply that by this column of PM and C. You just have to multiply that by this column, and then you're going to get this whole column here, and that's what this whole term is going to be here. So this entire term here is this going to be this. Maybe I should use a nice color. This part over here. This part is what this part here maps to. And then, so that's what each of these is telling you, that, you know, like if we were to say that M is 1 and C is 1, what would we get for this green? That's what this green is telling us would be like negative 0.01543. That's what the summation would be for M is 1, C is 1. That's what we would get here. And then for M is 1, C is 2, if we go across here, we would get, you know, for this part here would evaluate to 0.07790. Since it's a summation, we're just going to sum these values across, because these are all the values for M, all the values for C that are possible. So 1 to 4 for the bins for M, 1 to 4 for the bins for C. So we sum them together, and then we get our mutual information that we really want of 0.181919642. That's the mutual information between genes 2 and 3. So then you might wonder, okay, well, what about genes 2 and 5? So I want to just give another example as well. So again, like we do the same thing, and we the same kind of mapping where we have like genes 2 and 5, which we'd shown before, found the total count. You know, we kind of sum these values up here. These values, when summed, maybe this yellow. These values, when summed, will give us our total values here of 3.4 for gene 2. So these are actually like the same from last time, what you might observe, that is 3.4 and 3.4 is here. If you want to find gene 5 being in a certain bin, like let's say you want to find it like, you know, this 8.4, what we would do is, you know, for gene 5 being in bin 1, we would take this value here, we would take this plus this value plus this value plus this value, and then all together 3.1 plus 3.1, 6.2, 6.3, 8.4. That's how we get this 8.4. So it's the same thing from before, and we divide everything by 17.6, these counts, and we get these probabilities that the gene 5 is in this, gene 2 is in this bin, this is here. So that there should be the probability that you know, gene 2 is in this bin. So that's the probability that gene 2 is in this bin. So again, this is sort of like the probability that gene 2 is in this bin, probability that gene 5 is in a given bin. You know, like it has a really high probability, because, you know, I had 8.4 of being in bin 1, and then also like in bin 4 as well for gene 5. But then we can sort of see that this is sent to be 1 and 2, and then 3 and 4 has a slightly higher probability for gene 2 is, and then we found these counts and these values from before. And then we can just use the same formula as before, where, like, you know, same thing I used like as I did between gene 2 and 3, but here this is 1, and this is, you know, the same counts and probabilities, and then P of M and P of C, and then multiplying them together here. Multiplying them together here again, and this multiplication takes these two and multiplies them together. And then what this part is, you know, P of M and C, this part again is just going to be this here. PM and C is here. PM and C over here. PM and C, the joint. And this fraction just didn't show out, but it's just going to be here. That's a fraction. Actually, let me try to find that a little bit neater. So this fraction is here. And again, this part is yellow here, and this is here. So we can just sum them up together, you know, calculate this whole thing out, take the log 2 of this. So again, we have these probabilities, and we calculated these marginal probabilities, we multiply them together, get these values, then we divide this by this here to get this fraction. We take the log 2 of this cell here, you know, we take the log 2 of this cell to get this cell, and then we multiply this part by this part over here to get this value. Then we sum it up, and we get the mutual information between genes 2 and 5 is 0.570969. So we kind of showed another example for mutual information between genes 2 and 5. And, you know, I went over these two examples. You know, we went over genes 2 and 3, and then genes 2 and 5 as well. And we calculated the mutual information. using equal width binning, so going from here to the mutual information for these. But when you figure out the other examples, and I can make more videos going over those as well, what you're going to find again, like we had gone from this part to this part over here, equal width binning. But what you'll find if you finish up the other examples for the other genes is that this is the mutual information that you'll get for these pairwise genes. And then when you sort them, you're going to find that gene 1 and gene 4 have the highest mutual information of 0.721, and then genes 3 and 5 had the lowest of 0.156. And this was using the equal width binning. Again, please let me know if you get different results or any issues, but I recommend first calculating them, and then you can round them after you calculate them, sort these values, and then you can round them like to the nearest 3 decimal places. And what if instead we used equal density binning? So that was another approach. We had used equal width binning for this example, but what if we use equal density binning? So if you use equal density binning, you would get slightly different results here, and this is what you would get. So please let me know if you have any difficulties with this part, like kind of going from here all the way to getting the mutual information. And if you wanted to compare the results, between genes 1 and gene 4, it seems to be the highest here for equal width binning, but for equal density binning, it actually tends to be a little bit higher, which at gene 3 and 4. But where did this gene 1 and 4 end up being? Let's just trace it out. It ends up like going, let's see, where did he go? So what we can see is that this value, 0.721, so gene 1 and 4, it ended up over here. And what we see is 3 and 4. Well, where did those land up? Actually, it's a little bit comparable, but the numbers are different. So 3 and 4 actually ended up over here. You can sort of see based on the method you use for the binning, how you discretize it, you can get very different information. So that's sort of like showing you like how the method can play a role. And in reality, you would have like all the decimal places, and then you would just sort them, and then you could round them after you sort them, just if the numbers just so that they don't look too messy. So just sort of like tying in everything together in a way that we calculated the mutual information, and one of the things that now we want to do is we want to just remind ourselves why does this matter. Well, I mentioned that there is this RTN package that I use. It's on Bioconductor. It's really cool. It's an RTN package, to try to reconstruct transcriptional regulatory networks. So let's see, what did they say? So what they're using again is they are representing the relationships between variables, which are genes here. They're representing them with the mutual information value, and it can be positive or negative sign as well. And that's what they are sort of using, like we are trying to look for what is regulating FOXM1 gene. So they have found these transcription factors C1 OF106, HBB, MAPT, FSIP1, HBA2, and ZNF385B. And the key thing to know is that what happens here is that we are giving a gene expression data set, like sort of what we were looking at, except for time points we would have like different, it could be time points, or it could also be like stages, like in Alzheimer's or something like that, for instance. And then what we're getting is we're then going to also supply a list of transcription factors that we currently believe, and this package will then tell us, use mutual information and this arcane algorithm to try to infer using the mutual information. So for this gene, we're trying to figure out, okay, well, what we found is that we have, so this one is called, FOX M1. This is our gene of interest. And we're trying to then understand, okay, well, let's look and see, okay, then we have C1, ORF, 106. So these tend to be like OF proteins here, then this is HBB. Over here, that's what this part is telling us. Then we also have, this
FSIP. And then we're going to also have HBA2. And then we're also having this zinc finger protein that is ZNF385B here. And these are all transcription factors regulating this gene here. And so from here, we see that we have these corresponding, these corresponding values here that represent, the relationship between each of these transcription factors. These are transcription factors here and the target gene. And this is sort of like what we would put down for these values, like, the relationships that we're trying to infer here. And so again, just to reiterate, we have a gene expression data, it can be any, data like any set of genes. We were looking at these 5 genes, like gene 1, 2, 3, 4, 5, and I had come up with some examples, like in Alzheimer's, for instance. We have sample class labels. Those could be time points here. We're looking at time 0 to 150. This could also be like severe stage of Alzheimer's, or like first stage of cancer, second stage, and so on. And then for each pair of genes, we were trying to calculate the mutual information between them. And then we're going to use that information, So here they've given an example here where let's try to calculate the mutual information between gene I and gene J. That's what this will be here. And this information is going to be, what this thing is over here is kind of focusing on the mutual information between genes I and J. And then we're going to use that to infer, is there going to be an edge, like, or how strong is the edge in this, gene regulatory network. So here, normalized gene expression levels, you typically massage the data. Then you have the pairwise, mutual information that you're calculating. And based on that, you're going to try to, here you're trying to reconstruct, because there can be so many regulators, like thousands. It could be like Jaspers or, Lambert et al., from 2018. There could be their set of transcription factors. So you could have over 1,000, and in your gene expression data set, there could be a lot of them. So you just want to whittle it down to the ones that you believe are very, very important. And that's what this is going to return out of all of them. 1, 2, 3, 4, 5, 6 of these, 2, 3, 4, 5, 6. I might be missing one. FC1, or if I have AGB. Oh, I also found MAPT. Oh, yeah, this is actually huge. This is the microtubule associated tau protein. It's often associated with Alzheimer's. So we also just have to add this MAPT. Microtubule associated protein tau, which is really key in neurodegenerative diseases like Alzheimer's, where pretty much there is an accumulation of amyloid beta plaques between neurons and these tau proteins within neurons that disrupt like microtubule signaling and transport and functioning. So I just hope that this kind of makes sense, that we look across at all the genes, and some of these genes we kind of go in, we kind of figure out, okay, well, we think that some of these are going to be like transcription factors. So we sort of go in, we kind of label them here. So we see this GJ maybe and GK, the transcription factors. So that's what we tell RTN, that maybe we have some a priori, some information before, we run it, what we think could be transcription factors. And otherwise, it's like, it's also cool too, but sometimes those at prior can really help us, move in that direction and use that prior knowledge. Then using this mutual information, we can then construct the gene regulatory network, and that's going to be showing, how the transcription factors tend to interact with the genes that they're regulating to bring about the phenotypes of interest that we tend to observe in various diseases. And this just goes to show you how mutual information can be a key part in constructing gene regulatory networks and have a lot of applications even in computer science.
But and in reality, there's like you could have like all these genes and all these time values and points. But I just really hope that this helped. So if you were happy, you liked this video, please like my video, please subscribe to my channel, and please let me know if you have any questions. And I'll be happy to put up more examples on mutual information. So this is me, Sanya Kuller, and I'm really happy to help. And this is like concepts in computer science and advanced bioinformatics, and I'm just happy to help. So please let me know if there's anything I can do to explain this to you better. Thank you, guys.